Welcome back, everyone. As you can see, we are doing research methods, and this is a very important chapter, not just for AS psychology, but also for A2. You need to know your research method terms both in paper 1 as well as paper 2 in AS level, and similarly in paper 3 and paper 4. These terms will come in really handy. Paper 2 and paper 4, in fact, purely on research methods. So this is a very important chapter. We need to understand key research method terminology. We need to know how to apply them. And it's actually a really big chapter, so I'm going to split it into 3 sections. Today, I'm going to be discussing just some of the key terms. Then in another video, I will talk about data analysis, which is the stats part with the graphs and the mean, median, mode, etc. And then in a third video, I'll talk about the hypothesis and operationalization in more detail. For example, the one-tail, two-tail hypothesis. And once we're done with all of that, then I will make a video on how to answer P2 questions in general. And in that, I will also discuss the 10-mark design your own study question. So before we start, I've made like a list, or you can consider this a syllabus of everything that you need to know in research methods. So it's a good way to, it's kind of like a checklist that you have. So it's a good way to revise, to study. So you can just go over these points, you can go over these topics and just see what you do know, what you don't know. Because once you have all of this covered, then your research methods concepts are going to be pretty clear. So we have research method techniques, which include 6 types of studies that we can conduct: experiments, observation, self-report, case study, correlation, and longitudinal studies. What's important here are the definitions of each, the types of each, as well as the strengths and weaknesses of each. Then we have experimental design, which includes independent, repeated, and matched. Similarly, we need to know the definitions as well as the strengths and weaknesses of each. Controls and variables. This includes things like independent variable, dependent variable, confounding or extraneous variables, etc. Then we have 3 types of sampling methods. There are a few more which I will discuss briefly, but these are the 3 main ones. We should know the definitions and the strengths and weaknesses of all 3. Ethics for humans and animals, which, so because there are some studies which have animals as participants, but most of the studies have humans, so we can apply different ethical guidelines for each of these studies. Now, along with this, we have some key terms which we need to use, which are very important, which include validity and its different types, reliability and its different types, ecological validity and mundane realism. Now, you can argue that ecological validity is a type of validity, but I like to consider it a separate term because I recommend that students use it as a separate evaluative term. There's generalizability, demand characteristics, socially desirable responses, data. Now, data analysis, like I said, I will have a separate video on this, so I'm not going to be talking about this particular topic in today's class. Subjectivity, objectivity, experimental and control condition, random allocation, operationalization, standardization and replication will be covered when we do reliability. Order effects and its types, and how to overcome order effects through concepts such as counterbalancing, randomization. Aims and hypothesis, again, is a separate video: one-tail, two-tail, directional, non-directional hypothesis, and sample and population. So these are just some key terms which we should know. And when I do one particular topic, I'll be adding in these key terms. For example, when discussing experiments, I may bring in the definition of validity. When discussing variables and controls, I may bring in the definition of validity, reliability, etc. When talking about the sample, we'll be discussing, or sampling techniques, we'll be discussing what a sample is, what a population is, what a target population is, etc. So let's start. The first topic which we'll talk about is variables and controls. So by now, most of you are familiar with these terms: independent variable, dependent variable. But let's take a look at what are the exact definitions, because we should know the definitions, as this is something that can be asked in paper 2. An independent variable is a variable that is being manipulated to test for its impact on the dependent variable, and it has at least two conditions, which we refer to as an experimental condition and a control condition. You can refer to the independent variable and dependent variable as an IV and DV in your answers. You don't have to write the full form. A dependent variable is a variable that is being measured to test if the IV has an impact on it. So just by these two definitions, we can see that the two are related. They have a relationship, the IV and the DV, and the relationship that they have is one of cause and effect. And I'll explain this further with an example. The third type of variable are confounding variables. We can even refer to them as extraneous variables. And these are variables other than the IV that can affect the DV, and they lower validity. And within them, we have two types: participant variables, situational variables. And when we cannot control these, we call them uncontrollable variables. Controls is also a very important term that we should use and we should know the meaning of, because controls affect your study's validity and reliability. And controls are simply when we are able to reduce or remove the impact of confounding variables in our study. So having discussed the definitions, let's look at an example to really understand what all of these terms mean. Now, let me just test if my pen is working. Yes, it is. Okay. Now, let's say I am doing a study in which I want to test the impact of the air conditioning of a room, the AC, on students' concentration during a test. So I'm essentially conducting an experiment because, and I will discuss this in more detail when we talk about experiments, but an experiment is simply a study where we are testing the impact of an IV on the DV. Now let's understand what the term independent variable even means. A variable is a change, and an independent variable is a change that is happening that is being decided by the researcher. So when we say it is the variable that is being manipulated, it is the variable that the researcher is deciding as to how they want to change it. And the IV is basically the variable that is having an impact on another variable. So the other variable that is changing is dependent on something. It's dependent on the IV. So we call that the dependent variable. So in this particular example, we are looking at a change in concentration. So the change of the variable that we're looking at is concentration. The behavior that we're looking at is concentration, but we want to see if concentration is dependent on something. And the thing it is dependent on is the air conditioning in the room. So concentration becomes your dependent variable, the DV. Because if it is changing, it is not changing by itself, it's changing because of something. It's dependent on another factor or on another variable, which is why it's called a dependent variable. And the factor that it is dependent on is called the independent variable, the IV. So the IV is always the variable that is changing the DV. It is the variable that is affecting or impacting the DV. So just remember, in any question where there is an IV-DV, if you have to identify what the IV is or what the DV is, the behavior that is changing, the behavior that you're looking at of the participants is always going to be the DV. And the variable that is changing it, that is affecting it, is going to be the IV. So I'll give you another example. I want to see if lighting affects your productivity. So you're a worker who's working at an organization, and I want to see if different levels of lighting, bright lighting or dim or dull lighting affects your productivity. So productivity is the behavior of the participants that I'm looking at. So that is the dependent variable, but it is dependent on what? On different levels of lighting. So your lighting will be the IV. So whatever change is being brought about that is dependent on something is the DV, and the variable that is causing the change is the IV. So over here, the IV is the AC and the DV is concentration. Now, the dV is the variable that is being measured. So we are measuring concentration levels. And there are different ways in which we can do that, which I'll talk about later. But first, let's talk about the IV. The definition of IV, or independent variable, was the variable that is being manipulated. The variable that is being manipulated simply means that the researcher is deciding the different conditions of the IV that we want to set. So, for example, if I place people in a room where the air conditioning is on, and I give the students, let's say it's a classroom of 30 students, and I give them a test, and I want to see how they concentrate during the test. So the concentration is the dependent variable, and they're being tested in the room where the AC is on. And let's say that they are able to concentrate really well. And I want to conclude that, okay, air conditioning being on does affect your concentration. It does improve your concentration. But I need to compare it to another group that is taking the same test that does not have the AC on, just to draw a comparison. So if I have 30 students, let's say I split 15 students in one room and 15 in the other. So I have 15 students in the room where the AC is on, and I have another set of 15 students in the room where the AC is off. And I'm giving them the same test. So by doing this, what's happening is that I'm able to compare the impact that the AC has on the concentration of participants by comparing them in two different conditions, AC on and AC off. So by setting these two conditions, AC on and AC off, I am manipulating the IV. So when I say manipulating the IV, this is what I mean. Setting two or more conditions of the independent variable. So you've probably come across the terms different levels of the IV, different conditions of the IV. This is what we mean by setting two or more conditions of the independent variable, and the researcher can decide how they want to do this. This is completely up to the researcher. If I wanted, I could have also had two conditions where maybe the AC is at 24 degrees in one room and 16 degrees in another room. And then I can compare the results and see, are students able to concentrate better when the temperature is higher or lower? Okay, so the way in which we are setting the conditions of the IV is how we're manipulating it. Now, in this particular example, I have a case where the AC is on and I have a case where the AC is off. Now, keep in mind, what we really want to see is the impact of the AC on, right? Because it says the impact of the AC on concentration. So the group that is really being tested is the first group, this one, where the AC is on. We refer to this as the experimental group or the experimental condition, because this is the group on whom the experiment is actually being conducted. The IV is actually, the main IV is being tested on. The other group that is being used as a comparison is going to be called the control group or the control condition. Now, the reason why this is a control condition is because they are not being exposed to the IV, to the AC. They are just there as a comparison. They're taking the same test, so they're part of the procedure, but the IV is not really being implemented on them. We're not exposing them to air conditioning. We have the AC off for them. So they are just there as a comparison. Because if the 15 students in the AC on group are able to concentrate really well, and let's say they perform really well in their test, we need to compare it to the control group, and we need to see if these 15 students in the control group also perform really well. If they do not perform as well as the students in the AC on group, then we know it's purely due to the air conditioning and no other factor. So that is why we have to have two conditions, in which one can be an experimental group where the participants are actually exposed to that IV, and the other group is a control group where the participants are not exposed to the IV. So we can draw that comparison. Now let's take another example. Let's say the AC is on in both groups now. So again, I have 15 students in one class with the ACs on and 15 students in another class with the ACs also on. But what is the difference between these two groups now? In the first condition, I'll have the AC on at, let's say, 26 degrees. Whereas in the second condition, I'll have the AC at 16 degrees. So I'm setting a much lower temperature. Now I still have two conditions of my IV, AC on at 26 and AC on at 16. However, now because both groups are being exposed to the AC IV and there is no control group, I can say that I have two experimental conditions. But the difference between the two is how I've manipulated the IV by having the temperature higher for one group and having the temperature lower for the other. And let's say that the 15 participants, the 15 students in the room where the AC is at 16 degrees are able to concentrate much better and perform better in their test compared to the 15 in the room where the AC is at 26 degrees. I can conclude that a lower temperature helps people concentrate better. So that is the purpose of having two or more groups. In this particular study, I've given examples of two groups. If I want, I can have 3, 4, 5 groups. That depends on different researchers. But on average, it's most common that researchers set at least two conditions of an IV. So if you look at any study, let's say the Piliavin study, if you've studied that, one of the IVs is the condition of the victim, and it's manipulated in two ways. So two conditions are set, drunk or ill. So both of these are experimental conditions because they're both being exposed to a condition of the victim, but they're two completely separate conditions, drunk and ill. Another example is the race of the victim. So black, white. So they're both being exposed to different races, so which is why they're both experimental conditions, but again, there are two conditions. An example of a study with three conditions of the IV would be the Bandura study on aggression, where you have an aggressive condition where the model is aggressive, you have a non-aggressive condition where the model is non-aggressive, and then you have a third condition, a control condition, where there is no model present at all. So the reason that's a control condition is because the participant is not exposed to any model. And the reason why the other two are experimental conditions is because the participant is exposed to a model, but the behavior of the model is different in each. Similarly, if I wanted to have a third condition of the IV here, I could have a control condition where the AC is off. But just to keep things simple, I'll keep two conditions right now. AC on, and I'll stick with AC off. So 15 in each, and both are taking a test, and I'm assessing their concentration, and I'm going to see which group performs better. Now, if both groups perform really well, both groups are able to concentrate really well, then I can conclude that air conditioning does not have any specific impact on concentration, because then the control group would not be performing as well as the experimental group. So that is really why we have a control condition. Now, that's the manipulation of the IV. Let's talk more about the DV now. The DV is the concentration. Now, the definition of a DV is the variable that is being measured. So in this case, I'm measuring or I'm assessing concentration. Now, how can I assess or measure the concentration of participants? So one way is, as I'm doing in this study, by giving them a test. Because if students are able to concentrate better, they might perform better in the test. So their marks or their grades in a class test could be my way of measuring their concentration. Okay, so that's what I mean when we say we are measuring the DV. Now, let's say that I do this study and the students in the AC on group have performed much better in the test compared to the students in the AC off group. Okay, so my question is, can I conclude for sure that it is only the AC that is affecting your performance, your concentration? Okay, I'll repeat the question. The 15 students in the AC on group, let's remove this 26 because we now have AC on and AC off. So the 15 students in the AC on group, if they perform much better than the 15 students in the AC off group, can I conclude 100% that the reason for their better performance, their higher concentration, is solely because of the AC and nothing else? The answer to that is no. There can be other reasons as to why this particular group performed better than this group in the test, was able to concentrate better than this group in the test, other than the AC. So, what are these other factors that could affect the concentration apart from the air conditioning? Let's say noise level. Maybe there's a lot of noise outside the room of the students in the AC off condition, but there is no noise outside the room or around the room of the students in the AC on condition. So naturally, if there is a lot of noise pollution, students will not be able to concentrate. So maybe that is one reason why one group did better than the other. Another reason could be maybe the academic level. Maybe I split the participants in the two groups randomly, 15 each, and I happen to allocate more students in the AC on group who are just academically superior. They're better students, they have higher concentration levels in general, and the 15 who I put in the AC off group, they have generally a lower level of concentration. So again, it's not because of the AC, but instead it's because of their academic level. They're not good students, which is why they did not perform well as the students in the AC on group. So you can come up with many other reasons, many other factors, many other variables that can affect their concentration other than the AC. All these extra factors, all these extra variables that can also affect the DV, other than the IV, we refer to them as confounding variables or extraneous variables. So the definition, if you look back, was these are the variables other than the IV that can affect the DV, and they confuse the researcher. Now, as a researcher, I will be confused because I am only interested in the impact that the AC has on concentration, nothing else. I'm not interested in the impact that noise has on concentration. I'm not interested in on the impact of the test on your concentration. Maybe they're given two different tests. Maybe AC on participants are given an easier test. Maybe AC off participants are given a difficult test. Maybe that might affect their concentration. Maybe that might affect their performance. So I am not interested in the impact of these other variables on the DV, on concentration. I am only interested in the impact of the air conditioning. So what I need to do is I need to be smart about my study. And when I'm conducting my procedure, I need to be wary of these other factors. I need to think, okay, what are things other than the AC that could affect my DV that I'm not interested in? And once we identify them, we need to try and reduce their impact. We need to try and remove them or reduce their impact, or in other words, we need to try and control them. So you've probably come across this term many times in your classes where your teacher says the study has high levels of controls or so and so variable is controlled. This is basically what it means. It means controlling or reducing or completely eliminating the impact that confounding or extraneous variables may have on your study. So let's look at an example of how we can control noise. We can ensure that the two tests are taken at a time where there aren't many people around in the corridors or in the school so that there is no noise pollution. Or we can have someone outside each classroom to ensure that there aren't students disturbing or disrupting the classes and making a lot of noise. So noise is no longer a factor. Academic level now. So instead of splitting the participants randomly, what we can do is we can look at their academic records. We can see who's a good student, who's not, and we can split them into the two groups evenly. So an equal number of students with an A, B, C, D grade can be put in the AC on group, and an equal number of students with A, B, C, D grades in the AC off group. So now any difference in the results of the two groups cannot be due to their academic level because we've matched them in terms of this. Okay. Similarly, any other confounding variable that you can think of, You want to reduce, you try and eliminate them. You try to control them. Once I have controlled all these possible confounding variables, and then I conduct the study, where the only thing different now between the two groups is that one group has the AC on and the other has the AC off. And still, these 15 participants in the AC on group have performed much better than these 15 in the AC off. Then I can say, yes, it's due to air conditioning. Because now I'm not confused about any of these other variables affecting my DV as I've controlled them. So when I am sure that it is the IV that is affecting my DV of concentration, then I can say my results are accurate. And when I am sure that my results are accurate and that it is only the IV affecting the DV and not any other variable, I say that my study is valid. So this is what we mean by the term validity. And I know this is a term that a lot of students are confused about. It's something that students confuse reliability with. They keep asking me, what's the difference between validity? What's the difference between reliability? So this is basically what validity is. How sure you are that it is the IV affecting the DV and not any other confounding or extraneous variable. So in very simple terms, how accurately you are testing what you're supposed to without any other factor affecting your results. Okay, so that is what we mean by validity. Moving on to the slide about validity, here's the definition. And so these slides, by the way, are just for this presentation. I have detailed notes on the definitions of all of these terms. So if you wish to have access to those, my information, my contact details are mentioned on the very first slide. So please message me, please email me. I will be posting a link in the description below for a WhatsApp group that I've created. Many of you are already on it. And if anyone wishes to join, you can. I'll be giving regular updates about my videos on that group and any online classes that I may be keeping closer to the exam. So you are welcome to join and you're welcome to tell others to join as well. So do contact me and I can share my slides with you, my notes with you, the detailed notes on research methods as well as other content that I've created. Okay, validity is how accurately a study is measuring what it aims to measure. So the link between validity and controls is when we control confounding variables or extraneous variables, we are confident that only the IV is changing or affecting the DV. That is validity. Okay, so controls actually help increase validity because you're controlling extraneous variables, you're controlling confounding variables. And by controlling confounding variables, as a researcher, you are not confused about what variable is changing the DV. You are confident and you are sure that it is only the IV that is changing the DV, and that is what you want to test. That is your aim. I want to see the impact that AC has on concentration. So how accurately, how confident I am that I am testing the impact of just the AC on concentration, that is the validity of my study. Okay, so I hope you guys understand. If you don't, you can always message me separately and I can give you a separate, more detailed explanation. But hopefully this should suffice. Now let's look at other things that can also affect your validity. Demand characteristics and socially desirable responses. So these are two terms which we need to know, which can lower validity. Demand characteristics are basically when participants suspect the true aim of a study or suspect what is expected of them in a study and they change their behavior. So for example, I want to test the participants' concentration, but they are aware that I am testing their concentration. So what they will do is they will make a conscious effort to try and concentrate even harder. So they're changing their behavior. They are not showing their natural behavior. So it's not because of the AC that they're concentrating more or less. It's because they know that I am there for a particular purpose and they are changing their behavior accordingly. So whenever participants change their behavior on purpose and it's not due to the IV, that is referred to as demand characteristics. So they're not showing their natural, authentic, genuine behavior. So obviously, if you're not recording someone's genuine, authentic behavior, then your results cannot be accurate. And when your results are not accurate, your results are not valid. So that is why whenever there are demand characteristics in a study, we say that the study's validity is reduced. Now, there are ways to overcome this, which we'll discuss when we discuss the topic on ethics, because you've probably seen a lot of studies where participants are deceived. They are not told the real aim of a study because we don't want them to suspect the real aim. We don't want them to make an effort to try and change their behavior. We just want them to show their natural, authentic behavior to improve validity and reduce demand characteristics. Similar to demand characteristics are socially desirable responses. And socially desirable responses are pretty much the same thing, but um, there are certain research method techniques where participants are providing responses either verbally or in written form, which we refer to as self-report. So through interviews, through questionnaires, participants give information. And at times they can lie, they can give untruthful responses, or they can give an answer according to what they believe is socially acceptable. For example, the principal has come to your classroom and is asking you your opinion on your psychology teacher. Now, let's say that the entire class really likes the teacher, but you, on the other hand, do not like the teacher. Now, because all the other students have praised the teacher, rated the teacher really highly, when it's your turn and the principal asks you what your opinion is of the teacher, although you don't like the teacher, you might feel a certain social pressure to not answer according to your honest opinion. So instead of saying that you don't like the teacher, you will answer with a socially desirable response. You will say what everyone else said. So obviously, your answer is not genuine, it's not truthful. You've given the answer that you believe is socially acceptable, which is why, again, as a researcher, if I'm interpreting your responses, it is not accurate, it is not correct, it is not authentic, and hence it is not valid. Okay, reliability now. So another very important term, and another term that is affected by controls. So we know that controls are eliminating confounding variables, reducing confounding variables, hence allowing us to be sure that it is the IV affecting the DV, right? Now let's look at how controls affect reliability. So what exactly is reliability? One simple way to understand reliability is to know the word consistency. But if a question comes in paper 2 for, let's say, 2 marks, what is meant by the term reliability, and you simply write it means consistency, you will not get marks. Okay, this is just for your understanding. The definition that you should write is what's given over here. It is when a study has high levels of controls and it is highly standardized. It allows the procedure to be replicated or repeated in order to test for consistency or reliability of results. So controls are a very important word over here, and a few other important terms are standardization, which I've given the explanation of below, as well as replication. Now, every time we talk about reliability, we need to talk about controls. Reliability basically means, and let's take the AC example again. So let's say I've conducted the study and I have controlled as many confounding variables as I possibly can. Okay. Um, Now, by controlling all these different confounding variables and by assessing whether the students are in the AC on or AC off group, I came to the conclusion based on the results that students in the AC on room were able to concentrate better than students in the AC off room. Okay, so my study is valid and I published my study and these are the results. But what if this was just a one-time fluke? What if it was for this one particular study that I did, this one particular trial, where students performed better in the AC on condition or concentrated more in an AC on room as compared to an AC off room? What if in another situation, people are actually not able to concentrate that much when the AC is on, and in fact, they concentrate better when the AC is off? So, if that is the case, and I do not get the same results every time, then my study does not have consistency or my study does not have reliability. So reliability basically means, can you trust the study's results to always be the same way? So, and this is important because when I publish my study, institutes will use this information and apply it at their organizations or schools so that they can enhance the concentration of their students or their employees, for example. So when I publish my study, and let's say your school comes across it, your principal will think, okay, the study has shown that students concentrate better when the AC is on. So we'll apply this in our school and we'll make them take tests in an air-conditioned room. And hopefully, based on the findings of this study, the students will concentrate really well. Turns out that the students were not able to concentrate really well, and it had a detrimental effect on their concentration. So my study is not reliable. My study is not consistent. You cannot count on my study to always produce the same results if the results are not always producing the same findings as the first time I did it. So that is why reliability is important. So in order for me to test reliability of a study, I need to repeat my study. So when we say replicate a study, we mean to repeat it. But if I'm repeating the study, I need to do it the exact same way as I did it the first time. So the 30 participants who I split into two different groups, 15 in the AC on, 15 in the AC off, and I did the study by controlling all those possible confounding variables, and the 15 in the AC on were able to perform better or concentrate better. If I want to repeat the study to see if I get the same results, I need to do it the exact same way. So the same controls that I implemented the first time, I will implement the second time. So I'm doing the study again. I need to control noise again. I need to control the academic level again. I need to control the same test again. So I need to keep my procedure standardized, which means the same as it was for everyone the first way. And the only way I can keep it standardized is by implementing the same controls. So when I have high levels of controls, I am allowing for the procedure to be standardized, which means exactly the same for everyone. Everyone is taking the same test. Everyone has the same setting where there is no noise pollution. Everyone is at the same academic level. So through standardization, I can repeat the study or I can replicate the study for everyone the same way. And once I'm repeating a study more than once, I'm testing for consistency. I'm testing if I always get the same results every single time. So that is why controls affect reliability. Now, the studies in our syllabus, obviously, they're only being done once, which is why in our answers, we don't say that the study has high reliability, but instead we say that the study has high levels of controls. For example, this particular study controlled this variable, it controlled this variable. You give one or two examples of the controls that are implemented in a study, and then you can say that these controls make the procedure of the study standardized. And once it is standardized, we can replicate the study in order to test for reliability. So let's use an example of an actual study. Let's take the Andrade study, the doodling study. I can say that the study by Andrade has high levels of controls. For example, everyone heard the exact same mock telephone call, which was at 227 words per minute. It was 2.5 minutes long. All the doodling participants were given the exact same sheets of paper with the same number of circles and squares. All the control participants were given the exact same lined paper to ensure that they don't doodle. So I've given a few examples of controls from the study. Then I can say, this makes the procedure highly standardized and easy to replicate in order to test for reliability. If you write it as it is, as I just said, you will get full marks in your reliability evaluative point. So just to sum up, the difference between validity and reliability is, in validity, we are controlling confounding variables so that we are confident that it is only the IV affecting the DV, so that our results are accurate and correct. Whereas for reliability, we are implementing controls to make sure that the procedure is standardized for everyone so that we can replicate it or repeat it in future in order to test for consistency of results. So if you want to talk about reliability, we use controls in the context of replication and standardization. But if you want to use controls for validity, we use controls in the context of eliminating confounding variables. And speaking of confounding variables, there are two types of confounding variables which I forgot to talk about. These are participant variables and situational variables. Now, a participant variable is a confounding variable that is brought about by the participant. So for example, in the AC study, one of the possible confounding variables which we identified is the academic level of the student. Okay, so it is a confounding variable, but it's directly related to the participant. So it could be something about the participant, their personality, their intelligence, etc. So any confounding variable in a study that is directly related or brought about by the participant is called a participant variable. Another word for this is individual differences. The other type of confounding variable is called a situational variable. A situational variable is a confounding variable that is brought about by the situation. For example, the noise. If the participants are in a situation where there is a lot of noise and it's a confounding variable, then that would be a situational variable. Okay? When we cannot control a confounding variable, be it a participant variable or a situational variable, we call it an uncontrollable variable. So let's say that in our study, we did manage to control the noise, we did manage to control, let's say, what else? The academic level. Let's say the lighting of the two rooms are exactly the same so that lighting does not affect the concentration. So we managed to control all of these possible situational and participant variables. But let's say it's raining outside and the participants are getting distracted by the rain. Now that is not something I can control. So that will be an uncontrollable variable, and that will reduce the validity of my study. So it is not possible that you are able to control every single confounding variable in your study. Try to control as many as you can, especially the ones that you think will have a major impact on your study. And the ones that you are able to control will improve your validity. But the ones that you are not able to control can lower the validity, which is why for every study, when we talk about the validity or when we evaluate the validity, we tend to use validity as both a strength and a weakness. Confounding variables which are be controlled, we use those points as a strength for validity. But confounding variables which we are unable to control, or any possible demand characteristics or socially desirable responses which we are not able to control, those will reduce the validity. Okay. There are specific types of reliability: inter-rater and inter-observer reliability. So reliability, like I said, means consistency. Now, one research method technique which we'll talk about is observation. An observation is simply when you are observing the behavior of a participant. Now, when one researcher is observing the behavior of a participant, that one researcher can make mistakes in their observation. For that reason, a lot of times there are usually two observers who cross-check each other's observations to make sure that there are no mistakes. Very simple example. I am observing someone's aggression, and I noted that the person was punching the wall. Now, what if I made some mistake in my observation? Which is why I will have a second observer who will also observe the behavior of the participant. And if they also note the same observation, which is that the participant punched the wall, then I can say we have inter-observer reliability, because we both are observing the same behavior and we're reporting consistent observations that we observed of the participant's behavior. So that is inter-observer reliability. Okay. Inter-rater reliability is the same thing, but the only difference is instead of observing, we're rating the behavior. So I'm rating someone's aggression out of 10, and I observe someone behaving aggressively, and I'll give them an 8 on 10. I'll say this person is very aggressive, they're an 8 on 10. But obviously there could be a bias at my end, there could be some mistakes at my end. So the other observer will also observe the participant, and if they also give an 8 on 10, then we are sure that this person is actually very aggressive, because now two people are giving the same consistent ratings. Okay. An important point to note about inter-observer and inter-rater reliability is that the two observers should not know what each other are rating or observing, and they should only compare it once they've recorded their observations. So if I've given the participant an 8 on 10 in terms of their aggression, I should not know what my fellow observer has rated. Because if I know that he's also given an 8 on 10, then that might influence what I will rate. So if I completely by myself rate the person an 8 and the other observer also rates it an 8, and then we compare and we see what we rated what each of us rated, and we have the same or similar results, then we are sure that there is inter-observer reliability or inter-rater reliability. Okay. Split half method and test retest reliability are two forms of reliability which I will discuss when we talk about self-report questionnaires, because they're applicable to those techniques. Okay, here are some very important terms. Ecological validity. Ecological validity is simply the extent to which a study matches a real life setting. If you are doing a study that is close to an everyday life situation, for example, doing a study at a hospital or a school, we say that the setting is natural and hence has ecological validity. So by doing a study in a school where we're testing the impact of the AC on performance or concentration, we have high ecological validity, which is a strength. Now, along with ecological validity, I want my students to use the term mundane realism because it's part of ecological validity. Mundane realism basically means the task that we're making participants perform in the procedure. Is that something that they can expect to do in everyday life? Is it something that is normal or regular or ordinary and not something that is unusual? So, for example, in our study, the task that the participants are doing is taking a test in a classroom. Now, that is something that students can expect to do in their normal everyday lives. So I would say that because my study is being conducted in a natural setting of a classroom, it has ecological validity. And the task of taking a test in a room where the AC is on or off is something that students can experience in their everyday school lives. Hence, it has high mundane realism. Okay. So these are points that we use for our evaluations, especially when we talk about lab and field experiments. Another important point is generalizability. Generalizability is the extent to which the findings of a study can be applied to a wider population. If I'm doing a study on one person, my study does not have any generalizability because that one person is not representing anyone other than themselves. The behavior that that person shows is not applicable to others. However, if I have a larger sample, if I have 500 people, then I can say that my study has higher generalizability because now more people are being represented. Let's say I'm doing my AC study on 500 students, 250 males, 250 females, and they're all between the ages 17 to 19, and they're from 10 different schools in one particular city. So I would say that my study has high generalizability because my sample size is very large. Sample size basically refers to the number of participants. It's large, which is 500. That's a big number. It's also representing both male and female students, 250 each. So it's representing males, female, it's representing a larger population. It's also representing 10 different schools, so it's quite diverse. So you look at two things in generalizability: how large the sample is and how diverse it is. So this is a large sample and it is quite diverse. Males, females, people of different backgrounds, 10 different schools. So I would say generalizability is a strength, but I would back it up with all of these points. At the same time, I can also use generalizability as a weakness. How? You look at who is not being represented in the sample. They are all students from a particular age range, 17 to 19. So I cannot apply the findings to younger children or to adults over the age of 20, so 20 and above, or to old people, because they are not being represented in the sample. So my generalizability is not applicable to them. I also said that all the participants, even though they're from 10 different schools, are from one particular city. So maybe students in other countries, other cities, other cultures might behave differently. So I cannot apply my results or my studies results to students of other cultures or other cities as they are not being represented. So that is how we evaluate generalizability. We want generalizability to be a strength, but in one particular answer, we can use it as a strength and weakness. For example, in Piliavin, the sample size is very large of 4,453 participants. It includes an equal mix of a racial mix of white and black people, 55% white, 45% black, and it includes both males and females and people of different backgrounds. So quite diverse, large sample, all of these points make it representative of a larger population, hence generalizability is a strength. However, these are all people traveling the New York subway. They're all from New York, so the sample is not representing people of other cultures, where helping behavior might be very different compared to what we saw from these people. Hence, the findings cannot be applied to people outside of New York, which lowers the generalizability. The generalizability in one answer for one study can be both a strength and a weakness. In some studies, it is only a strength, where it is a very large sample, where a very large number of people, as well as a very diverse set of people, are being represented. And in some studies, it is only a weakness, such as a case study where there is just one participant. In some studies, it is more a strength and less so weakness, and in others, it is more so weakness and less so strength. Like in the Pilyavin study, it is actually more a strength than it is a weakness. Here's another term, order effects, and this is something which also reduces validity. It can technically be considered a type of confounding variable. Okay, so anything that reduces validity is technically a form of confounding variable. Order effects is when the order of a task affects the behavior of the participants. So, what does that mean? It means, let's say, I am showing you two paintings, a red painting and a blue painting. And I want to see if the color of the painting has an impact on your memory. So are you able to remember details better of the blue painting or the red painting? So over here, the IV would be color and the DV would be memory, because the behavior that you are assessing is the memory of the participants. But what is it dependent on? It is dependent on the color of the two paintings, so blue and red. So color of painting is the IV, and the thing that it is changing or affecting is your memory. So memory would be DV. And the color has two conditions that the researchers manipulated. So the two conditions of the IV would be red and blue. So blue painting and a red painting. Okay. So that's the IV and DV. Now, let's say that I show you the red painting first for 5 minutes, and then I show you the blue painting for 5 minutes. And then I give you a 1 hour break, and then I ask you to try and recall details of each painting. And it turns out that you were able to recall details of the blue painting a lot more than details of the red painting. So I will conclude that blue helps people remember better than the color red. But maybe that's not valid. Maybe those results are not accurate. And the reason is, maybe it's not the color that helped you remember, but instead the order in which I showed the paintings. So order effects could have perhaps hindered the results. Now, how would the order affect your results? I showed you red first, and then I showed you blue later. So maybe because you saw blue more recently, it is fresher in your mind. So you are able to remember more details from blue compared to red because you saw blue more recently. So it's not because of the color, but instead because of the sequence or the order in which I displayed the two conditions of the IV, the two paintings to you. So that reduces validity because, again, when I conclude my studies results, I will say blue helps people remember better than red. But in reality, that may not be true. And the reason your results were the way they were is simply because of order effects. Okay, so that's why it reduces validity. Now, there are two types of order effects: practice effects and fatigue effects. Order effects could also be when you perform a task multiple times. Okay, you're doing the task once, then again, and again. So the fact that you're doing the procedure in such an order that you're doing it multiple times, you can memorize what you have to do. So again, let's say that the two paintings, red and blue, are being shown to you, and they're the exact same paintings. The only difference is the color. One is painted in red and one is painted in blue, but they're the exact same painting. So the first time that you see the painting, you'll probably try to remember a few details. And the second time you look at it, you already know what you have to do. You already remember details from the first time you saw it. So you've already memorized some details. So the second time you will obviously remember it better because you've already seen it once. So this is practice effects. So again, it's not because of the color, it's the order. The fact that you've seen it once, you've done the procedure once, that you've memorized what you have to do. Let's give you another example. Let's say that you take a test, and let's say that you get 7 on 20 in the test. It's an MCQ test. I give you the same MCQ test the next day. Now, it's a psychology test. Let's say it's an MCQ test on research methods. Now, when you get 7 out of 20 the first time, it tells me that you haven't studied and you're not a very good student. Now, I give you the same MCQ test on research methods the next day. If you are genuinely not a good student who hasn't studied, you should get around about 7 or 8 again out of 20. But if the second time you end up getting, let's say, an 18 out of 20, I will be confused because I don't know if you are a good student or a bad student. Maybe you didn't study the first time, you did the second time. Or is it because you've just memorized the answers from the first time that the second time you've already practiced the test once, so now you know the answers? So if that is the case, then you have succumbed to practice effects. So, okay, so a type of order effect where you've done the procedure more than once, so you now are better at it because you know what the answers are, you know what you have to do. So because of practice effects, if you do end up getting an 18 the second time, it is not a valid representation of how you are as a student. It still doesn't tell me that you studied really well. I may interpret it that way, but it would be an incorrect interpretation. It would not be valid because you've only done better because you've memorized what you had to do. So those are practice effects. The other type of order effects are fatigue effects. This is when you do a task more than once and you become tired. So maybe you actually are a good student, and let's say you get 15 out of 20, and you did study, and I gave you the exact same test the next day. But the next day you're tired because you already took the test, but you studied really hard for the test one day before. So the next day, back to back tests, maybe you're really tired. So this time, the second time you take it, your performance is not as good. And the second time you get, let's say, a 5 or a 6 or a 7. So again, it's not a valid representation of how you are as a student. It's only because you were tired or fatigued or maybe even bored the second time that you did not do as well. Another example, let's say you're very good at swimming and I want to assess your performance in swimming, and you've been swimming for 5 to 6 hours straight. And then after 5 to 6 hours, I come to assess your performance and you're very tired. The fact that you've been doing the task again repeatedly for so long, for so many hours, when I'm assessing your performance after 6 hours, naturally you're going to be tired, you're going to be fatigued, and your performance will not be as good as it would be had you been fresh. So when I'm observing you, when I'm interpreting your performance in swimming, I will think, this person is not a very good swimmer. They've not got a very good time. So that would not be a valid interpretation of your behavior because you are succumbing to fatigue effects. So these are order effects and they lower validity. Now we need to talk about how we can overcome order effects. One way is a process called randomization. So randomization would be that you are randomly displaying different um, stimuli to a participant so that the order is not affecting their performance. So let's take an example of this. So there was a study which has now been removed from the syllabus. It was by a researcher named Canley. And what Canley wanted to do in his study is he wanted to see if people find, if people will remember people will rate negative images as being more emotional or neutral images as being more emotional. So a negative image could be a picture of someone being burnt alive or someone being tortured, whereas a neutral image could be a picture of a tissue box, because there's a tissue box in front of me, so I'm using that example. Now, the participants would have to rate from 0 to 3 whether they found the picture emotional or not. Okay? 0 being not at all emotional and 3 being extremely emotional. And there were 96 pictures that were shown to the participants. And from these 96, 48 of them would be negative pictures and 48 of them would be neutral pictures. Now, if the participants are being shown all 48 negative pictures first in a row, the first picture they see, they look at it and they'll think, okay, this is an emotional picture, this is a negative picture, I'll rate it a 3. Then the second picture is also a negative picture, they'll rate it a 3. Third one, fourth one, fifth one, sixth one, all are negative pictures, they'll rate them 3 on 3. Eventually, maybe by the 10th or the 12th or 15th picture, they'll start thinking to themselves, hold on, all of these pictures are negative. And after a while, they may not even focus on the picture anymore. And even before looking at the picture, when the next one is displayed, they'll automatically rate it a 3. So they're not even focusing on the picture. So their interpretation, their analysis, their ratings on the picture is not going to be valid because they are showing practice or order effects. Because they now know that it's always going to be a negative image, so there's no point looking, let's just rate it a 3. So to overcome this, we can randomize the sequence in which we are going to show the pictures, which is what Canley did. So rather than showing all 48 negative pictures first and then the 48 neutral pictures, Canley actually randomized them so that at any point in time from the 96 images shown, any one of them could be a negative picture or it could be a neutral picture. So now the participant actually has to look at the picture, focus on it, and then rate it. Because maybe the first two could be neutral, and then the third one could be negative, and then the fourth one could be neutral again. So now you cannot rate based on practice effects. Now you actually have to look at the picture, analyze it, and then based on what you feel, you will give it a rating of either 0, 1, 2, or 3. So that is randomization. That is one way of overcoming order effects. Another way of overcoming order effects is a concept called, or a technique called, counterbalancing. The definition of this is when we do two tasks, let's call them task A and task B, in the order A, B, and then B, A. Now, let's go back to the painting example. Let's say that there are 10 participants who are part of my study, and I want to show them these two paintings, the red painting and the blue painting, and I want to see if they're able to remember details of the blue painting more or the red painting. Now, I don't want their results to be affected by order effects. So what I'll do is, I will make half of the participants, so 5 of them, see the red painting first, and then the blue painting. And the other 5, the other half, I will make them see the blue painting first and then the red painting. So the other way around. So I'm counterbalancing the task. So the first 5, they will see the blue painting first, then red, and the other 5 will see the red painting first and then blue. And if both of them still recall details of the blue painting more than the red painting, then I know it is because of the color and not the order, because I have counterbalanced the order. So it can't be because of the order that one has seen it more recently than the other, because half of them are seeing red first then blue and the other half are seeing blue first then red. So now I know it is because of the color and not because of the order. So this is a way of counterbalancing a procedure to overcome order effects. A third way is through an independent measures design, but for this we have to do experimental design, which I will discuss later on. Okay, data refers to the findings of a study, and every time we do a study, when we are finished with the procedure, we get results and we analyze the results to see whether the aim that we were testing was supported or not. Now, these results, this data can be quantitative and qualitative. Quantitative data refers to data that is objective and numerical or statistical, and it allows for comparisons to be made. It basically tells you what the results of a study are. So let's take, for example, the Milgram study. There were 40 participants. 26 out of the 40 participants administered the maximum 450-volt shock and were labeled obedient, whereas 14 out of the 40 did not go up to the maximum 450 and they withdrew before administering the maximum shock and they were labeled as defiant. So this quantitative data is allowing us to compare different participants based on the level of voltage that they administered. In our AC study, The reason we are able to determine that students in the AC on, let's say, perform better or were able to concentrate better than students in the AC off is by looking at the grades that they get in a test. So those numbers, those grades is basically objective or quantitative numerical data. So we are comparing two different groups. So that's quantitative data. It tells you what the results are, but it doesn't tell you why the results are that way. Why is it that the 26 participants went to the full 450 shock, whereas the other 14 did not? Why is it that the students in the AC on performed better than the students in the AC off? Is there any other reason apart from the AC? That cannot be figured out through quantitative, but it can be through qualitative data. Now, qualitative data is data that is detailed, in-depth, and subjective, and it is in the form of comments, opinions, and emotions or behaviors of the participant. So if a student in the AC on group has performed really well, I can ask them, what is the reason? I can ask them, did you feel comfortable in the AC? And let's say that one person says, yes, I felt really comfortable. I was able to concentrate really well. So that tells me that it is because of the AC. But another student says that, no, it wasn't the AC. It's because I cheated in my test. So through qualitative data, through comments and opinions of participants, we can actually understand why the participants behaved the way that they did. So we can understand the reasons behind their behavior. That's why we say it's detailed, in-depth, and subjective, because it's different from person to person. That's what subjective means. It doesn't have to be the same for everyone. So that's qualitative data. So anytime any study has comments, behaviors, emotions, we call that qualitative. But when we have numbers, numerical data, statistical data, which is objective, we call it quantitative. Okay, a big and important topic is ethics. Ethics are the moral and legal guidelines that researchers should follow when conducting studies on participants, be it either humans or animals. So a researcher cannot just do whatever they will. There are certain guidelines that they should follow. However, most studies you've probably seen by now do not always follow ethical guidelines, and there are reasons for that. So let's look at the ethical guidelines that we need to follow for human participants. The first is deception. Now, deception basically means lying to the participant about different things. For example, the aim of the study. For example, the Milgram study, the participants were not told the true aim, they were given a fake aim. So they were deceived about the aim. So that is unethical. We want a study to be ethical, so we should tell them the real aim. We should not deceive them. But the reason why participants are often deceived is, going back to demand characteristics, to avoid participants from changing their behavior, from suspecting the real aim and showing demand characteristics. Because if the participants, let's say let's take our study, the AC study, if they know that the study is on assessing their concentration, they might make a conscious effort to try and concentrate harder. So it won't be because of the AC, but instead because of the fact that they know what the aim is. So if we deceive them, we give them a fake aim, or we don't tell them the aim, there's a greater chance that we will not have demand characteristics and the study's results will be valid. So it will be valid, but at the same time, the counter argument is that it will be unethical because we are deceiving them. So we kind of have to pick and choose at times. Do we want to increase validity and avoid demand characteristics at the cost of breaking ethical guidelines, or do we want to be ethical and not deceive them, but at the same time risk the chances of demand characteristics which may lower validity? So we can deceive them in other ways also. Within the procedure, somehow we can deceive them. If you're using a questionnaire, self-report, we can give them fake questions, filler questions about a fake aim. We can even use a researcher who can pretend to be the participant or pretend to be someone other than the researcher to try and influence the behavior of the participants. For example, the victim or the model in the study by Piliavin, or the learner in the study by Milgram. So when a researcher is part of the study and pretends to be a participant, we refer to them as a stooge or confederate or actor. This is also a form of deception because the participant does not know that they are actually part of the research team. So these are just different ways in which we can deceive the participants, but it will be unethical. Informed consent refers to taking permission from a participant before doing a study on them. I cannot do a study on you without your consent. So I need your informed consent. At times, participants do give consent, but we don't tell them the real aim. So they're giving consent for a fake aim, and technically they haven't given consent for the real aim of the study. Again, Milgram is an example. Participants gave consent, they gave permission to be part of the study, but they thought they're taking part in a study that is testing the effects of punishment on memory and learning. They did not know that we're actually just testing them on their obedience. So technically, even though they gave consent, it wasn't informed consent because they were not informed of the true aim. So you can say that informed consent in that case was broken. Right to withdraw is basically participants have the right to exit the study when they wish. We cannot force them to continue against their will. If they feel they're going through discomfort or they're feeling uneasy or going through any potential distress, we should not force them to continue and they can exit the study in the middle. Confidentiality means maintaining the personal information of the participants, such as their identities, where they're from, because at times participants may be embarrassed by their behaviors and they don't want their identities to be revealed. So when publishing studies, we should keep their personal information confidential. Similar to confidentiality is privacy. We should make sure that we are not asking participants any really personal or private questions about their personal lives that may make them feel uncomfortable. Because if we do that, we would be causing them psychological harm. And this brings us to the sixth point, which is protection from harm. During our procedure, we must ensure that participants are being protected from any physical or emotional or psychological harm. But again, this may not always be the case. And I go back to the Milgram study. So by now you've probably realized that the Milgram study does have a lot of ethical issues. It's not a very ethical study, but it is a very valid study. And in that, the participants went through a lot of short-term psychological harm. They were sweating. Some participants even experienced seizures. So they were not protected from psychological harm. So that was unethical. And lastly, debriefing. Now, debriefing is when we reveal the true purpose of the study. We tell them the true aim and we tell them everything that the study is actually about. Now, if we don't wish to deceive the participants, there is no need to deceive them. We can debrief the participants at the beginning of the study. However, at times, if we do deceive participants to avoid demand characteristics, to kind of counter that, at the end of the study, we can debrief them. We can let them know what the study was really about. And Milgram did that. Milgram deceived them throughout, but at the end, he debriefed them. And another reason why it was important to debrief participants is because if you're causing them any psychological harm during the study, you want to make sure it is only short-term harm and not long-term harm. So by revealing the true purpose of the study, by, for example, in Milgram, making them meet the stooge and ensuring that he was not harmed, the participants did get some sense of relief. So by debriefing them, they are aware of the true aim, and we can also try and avoid any long-term psychological harm. Now, when we have animals as participants, there are ethical guidelines that are different from those that we use for humans that we need to consider. The first one is numbers. Try and use the minimum number of animals possible to achieve your study, the aim of your study, in a valid and reliable manner. Because in most cases, we'll be doing a study on an animal outside their natural habitat, such as in a lab. So it can be distressing for the animal. So try and use the minimum number of animals that you possibly can. If you can conduct a study in a valid and reliable manner on 2 parrots, on 2 chimpanzees, 2 monkeys, let's say, there's no need to have 10. Okay. If you have the option to replace the animal with, let's say, video footage of other similar studies or computer simulations, then consider that. Okay, so that's the guideline of replacement, rather than putting them in a lab and out of their natural habitat. Similar to psychological harm, you should ensure that we're not causing any pain or distress, emotional or physical, to the animal during the procedure. Make sure that you're using the correct species of animal appropriate for the study so that there is no strain or distress caused to them. Now, during the procedure, if the animal is showing any desired behavior, make sure you're rewarding them. Make sure you're giving them a toy or something that they desire to keep them motivated. And do not deprive them of their basic needs like food, water, or safety and shelter. Aversive stimuli basically means aversive is something you wish to avoid, and stimulus is anything that causes a reaction. So do not put anything in the animal's environment that may cause the animal to have a negative reaction, which basically means do not put anything in the animal's environment which may threaten it or make it feel scared or harmed. So, for example, if you're doing a study on rats, don't put them around cats because they would feel threatened by them. So, cats would be an aversive stimuli. So, this is similar to the point of pain and distress. So, try to avoid pain and distress by not putting any aversive stimuli or stimulus in the animal's environment. And lastly, housing and caging. This refers to the way in which we keep animals in the procedure, how we house them. Some animals are social animals, so rather than keeping them isolated, we should keep them in groups with animals of the same species so that they don't feel isolated or alone. And caging refers to how we keep the animals in the procedure. Are we keeping them in cages? If it's a cat or dog and there's no need to keep them in a cage all day long, then we shouldn't. Even if we keep them in a cage, we should make sure that they're not kept in overcrowded cages or with other animals that may harm them, or there is sufficient space for them to move around and they're not kept in very small spaces. So these are some animal ethical guidelines that we should keep in mind when conducting studies on animals. And I believe there are two animal studies in our syllabus. HACCP, which is on monkeys, and FAGAN, which is on elephants. Okay, let's get to one of the most important points about or topics in research methods, which is the research method techniques. Now, this is very important because these are the different ways in which we can conduct psychological research. Okay, if you're a researcher and you have an idea in your head that you want to test or an aim that you want to test, you can do so by acquiring participants and conducting a procedure, conducting a study. Now, the way you conduct the study is basically called your research method technique or your research methods. And there are 6 that we will discuss. The first is experiments. What is the definition of an experiment? An experiment is a study in which the independent variable is manipulated to test for its impact or effect on the dependent variable. Now, we've already discussed what an IV and DV is. An experiment is a study that has an IV and DV. Remember that. None of the other research method techniques have an IV or DV. The only way these other research method techniques such as observation, self-report, case study, etc. would have an IV-DV is if they are being used along with an experiment. So for example, we're doing an observation within an experiment, like Piliavin. But the IV-DV aspect is not because of the observation, it's because we're doing an experiment as well. So any study in which we are testing the relationship of cause and effect between two variables, or in other words, where we're testing the impact of an IV on a DV, we call it an experiment. Okay, so the FAGAN study, elephant learning, is considered a controlled observation, not an experiment because there is no IV-DV. The study by Saavedra and Silverman, button phobia, is a case study. There is no IV-DV. But the study by Bandura is an experiment because there is an IV and there is a DV. The study by Piliavin, Milgram, well, Milgram not so much because that is something I'll talk about when I upload the Milgram video. But Piliavin has an IV, has a DV, so that's an experiment. Andrade has an IV-DV, Baron Cohen has an IV-DV, so these are all experiments. And there are 3 types of experiments: lab, field, and although this has been removed this year in the textbook, I will still cover it because we do have a natural experiment in our syllabus in the form of Baron Cohen, but it's also considered a lab experiment. But I'm including it in the research methods because it is important. So we have lab, field, and natural experiments. A lab experiment is a study, an experiment conducted in an artificial and highly controlled setting of a lab. But if a question comes in paper 2 on the definition of a lab experiment, you should mention these exact words, which is a study in which the IV is being manipulated to test for its impact on the DV in the artificial and highly controlled setting of a lab. Now, if you write it's a study which is conducted or an experiment conducted in the artificial and highly controlled setting of a lab, that might be okay, but what you're essentially doing is just describing lab, not really describing experiment. So by saying that it is a study in which the IV is being manipulated to test for its impact on the DV, we've described the experiment aspect of the term, And then by adding in the artificial and highly controlled setting of a lab, we're adding the lab aspect of the term. So for lab experiment, we should mention both these points. Field experiments are experiments in which the study is testing the IV's impact, manipulating the IV to test for its impact on the DV, but in a natural setting. So the difference between a lab and field experiment is the environment in which we're doing the experiment. If you're doing it in a highly controlled and artificial environment of a lab, it's a lab experiment. But if you're testing the impact of the IV on the DV in a natural environment, it is a field experiment. Now, what do we mean by a natural environment? A natural setting is basically any setting where any setting which you can consider to be an everyday, ordinary life setting. So outside the lab, basically a hotel, a hospital, a classroom, a school, a subway, an airport, a train station. So even your house can technically be a natural setting. So any experiment that is being conducted in an everyday, ordinary life setting is considered a field experiment. Okay. Keep in mind the difference between lab and field is the setting, artificial setting compared to natural setting. Natural experiment has nothing to do with the setting. This is very important because students end up confusing field with natural experiment all the time. They see the term natural experiment and they think, oh, natural setting. So a field experiment is natural setting. A natural experiment has got nothing to do with the setting, but it is when the IV is naturally occurring. So we can do a natural experiment in a lab, we can do a natural experiment in a field setting, that doesn't matter. As long as the independent variable is naturally occurring, we call it a natural IV. Now, what do I mean by naturally occurring IV? It is an IV which cannot be manipulated. So this is why at times natural experiments are also called quasi-experiments or half experiments, because the true definition of an experiment is one where there is an IV which can be manipulated to test for its impact on the DV. But when you have an IV and it cannot be manipulated, then it's technically a half experiment or a natural experiment. And let's say, I'll give you an example now of an IV that cannot be manipulated. Let's say I want to conduct a study in which I want to see the effect of the weather on your mood, weather on your happiness. So, and the two conditions I've set are rainy weather and sunny weather. So, I'm going to assess your happiness when it's raining, and I'm going to assess your happiness when it is sunny. Now, the problem is, as a researcher, I cannot manipulate the weather. I cannot decide, okay, today it will be rainy and then tomorrow it will be sunny. I have to wait for this to happen naturally. So it's not in my control. I cannot change it by myself. I cannot manipulate it myself. So this, although it's an experiment, it's an IV, sorry, because it's a variable that is affecting another variable, which is that of your happiness. It is not something that I can manipulate, which is why it's a naturally occurring IV. Which is why the Baron-Cohen study is also considered a natural experiment, although it's predominantly mainly considered a lab experiment. It's called a natural experiment because the participants either have autism or they don't. That's not something that you can manipulate. You can't say, okay, today you will be in the autistic group and tomorrow I'll put you in the non-autistic group, right? It doesn't work that way. It's a naturally occurring IV. Now, having covered the definitions, let's talk about the strengths and weaknesses of each. So let's talk about lab experiments first. And it's important to know the strengths and weaknesses because these are the main points that you tend to use for your evaluations in paper 1 for 10 marks. Now, one strength of a lab experiment is we know that it's a highly controlled setting, right? We saw that in the definition. When we say highly controlled, we're basically saying it has high levels of controls. So when we have high levels of controls, we already discussed what controls can do. Let me just write this in a bigger handwriting in case you guys can't see it. So lab experiment, we're talking about the strengths. The first strength is it has high levels of controls. Now, having discussed the topic on controls, we know that controls affect two things, validity and reliability. So one go-to strength of any lab experiment is that because there are high levels of controls, it makes the procedure standardized. Everyone is going through the exact same controlled procedure. And when it is standardized, we are basically replicating or repeating the study the same way for everyone. So easy to replicate. And when we replicate a study, we are essentially assessing its reliability. So reliability is a strength of lab experiments. Any lab experiment in your syllabus, when you're evaluating it and you use reliability, you will always get full marks. Just make sure you use these key terms. High levels of controls, making the procedure standardized, allowing it to be replicated easily to test for reliability. Just make sure that you give a few examples of the controls from the study. Okay, everything that was kept standardized or controlled in the procedure, give one or two examples, and then you can write the rest of the statement, and reliability will be your strength. Another strength, again, because there are high levels of controls. Now remember, when you have controls, what are you reducing? What are you eliminating? You are reducing confounding variables or extraneous variables. And when you are reducing extraneous variables, what does that mean? It means you are more confident that only the IV is affecting the DV, hence increasing validity. So validity would be another strength. Of course, there can still be some points which can lower validity because of confounding variables that you are not controlling. But overall, generally, a lot of confounding variables are controlled. And just give a few examples from the study where confounding variables are controlled. One or two examples should suffice. And then you can say this allows us to be confident that the IV of so-and-so is affecting the DV of so-and-so, hence increasing validity. A third strength is, now look, as a participant, you don't just happen to show up at a lab by chance, right? You've probably volunteered for the study, you've probably given consent, you know you're there for a study. So you've given permission to be part of a study. That's why you're there at the lab. So in most cases, there is a chance that it will be ethical in terms of informed consent. So informed consent is usually there, which makes the study ethical. Again, this depends from study to study, because if you've deceived them about the real aim, like Milgram, then technically there is no informed consent. So this is not a go-to strength. It depends on the procedure of the study. It depends on whether there is consent or not. But in most cases, yes, there is. And the fourth strength is data is collected, which is quantitative. In any lab experiment, you will collect quantitative data, which is objective, numerical, give a few examples of the data from the study, and then you can say it allows for comparisons to be made. By giving examples, by giving numbers, you can make those comparisons. So data is basically going to be your fourth strength for lab experiments. Now let's talk about the weaknesses of a lab experiment. Now keep in mind, these 4 points are not the only points that you can write in your evaluation, but these are the 4 points you can consider if your evaluative point is about lab experiments. Of course, you can use other points as well in your evaluation, such as generalizability, such as ecological validity, such as the experimental design, the sampling method, etc. But those are points about the study, not specifically about lab experiments. So what are some weaknesses? The number one weakness of a lab experiment is that it has low ecological validity because it is being done in an artificial setting of a lab. So like I said, one go-to point for strength is reliability. One go-to weakness for any lab experiment is ecological validity because it's being done in an artificial setting, it's being done in a controlled environment. Therefore, it does not have ecological validity. And in most cases, and you will refer to the procedure over here, in most cases, the task or the procedure that the participant is taking part in a lab experiment is not something that they perform in everyday life. For example, in Milgram, it's not common for people to be giving electric shocks to each other for answering incorrectly on a word pair task. So mundane realism is also usually a weakness and low in lab experiment. So you can use these two terms together as a weakness. Another potential weakness is, and this is going back to the point about consent. So we talked about how if there is consent, it is a strength because it is ethical. But remember, if participants are aware that they're part of a study, if they've given consent, they know that they're part of a procedure, it may lead to demand characteristics. Again, there is no guarantee. It depends on how well you've hidden the aim of the study. But if there is complete consent and the participants know what the procedure is about, they may show demand characteristics which can lower validity. But this, again, like I said, depends from study to study, because like there was consent in Milgram, but not for the real aim. So there were no chances of demand characteristics there, which is why validity is actually a strength. So these are your two weaknesses for a lab experiment. And again, like I said, these aren't the only weaknesses that you can use in your evaluation. You can use other points, but these are your lab experiment related weaknesses that you can refer to. Okay, now let's talk about the strengths and weaknesses of a field experiment. It's actually the opposite of a lab experiment. So for a field experiment, one strength is ecological validity and mundane realism is high, because it is a natural setting and the task is usually something you expect in everyday life. Like in Pilyavan, it's being done at a subway, which is an everyday life setting, and you're helping someone who's fallen down, which is something you can experience in your daily life. So mundane realism and ecological validity would be a strength. You get a lot of qualitative and quantitative data. So qualitative data is usually gathered along with quantitative. So you can give both points, you can give examples of both and talk about how both are useful. In field experiments, participants may not know that they're part of a study. There might not be consent, so more chance that the participants will not show demand characteristics. So if we don't have demand characteristics, then that will increase the validity. So these are your strengths of a field experiment. Let's talk about some weaknesses. So one weakness that we can consider of a field experiment is when we compare it to a lab experiment, there are fewer controls, because the fact that you're doing a study in a natural environment means that there may be a lot of variables that you cannot control, situational variables, participant variables that are out of your controls. So because of that reason, if there are any uncontrollable variables, it affects two things. It can affect your validity. So there may be a chance that your study may lack validity if there are high levels of confounding variables that you were unable to control. And at the same time, because if you have fewer controls compared to a lab, it is less standardized than a lab experiment, more difficult to repeat the study in the same standardized way, so more difficult to test for reliability. Now, this does not mean that reliability or validity is a weakness of field experiments. It depends on the nature of the procedure. If you have controls like Pilyavan, even though it's a field experiment, it did have many controls. So reliability and validity are not as much a weakness as they are a strength. But generally for field experiments, compared to a lab experiment, you can expect fewer controls. And so as a comparison, you can expect lower levels of validity and perhaps even more difficult to replicate for reliability in field experiments. And the second problem or the second weakness is, again, going back to the point about participants perhaps not knowing that they're part of an experiment. If that is the case, then it'll be unethical because they technically would not have been providing their informed consent. So these are your strengths and weaknesses for a field experiment. Now, for a natural experiment, you don't really have too many strengths weaknesses of its own because it depends on where the study is being conducted. Because like I said, you can conduct a natural experiment in a lab or in a field environment. It's just that the IV is naturally occurring. But one aspect of a natural experiment that you can maybe consider a strength is that because the IV is naturally occurring, maybe it adds a little bit of ecological validity because that is something natural. That's a natural element that you're bringing to the study. But the weakness at the same time is the fact that it can be difficult to replicate. So if I want to test the impact of the weather on your happiness, I need to wait for it to be rainy. I need to wait for it to be sunny. It's not something I can manipulate myself. So the next time I wish to repeat the study to test for reliability, I'll have to wait for it to be rainy or sunny again. So it can be very difficult to replicate um, a natural experiment for that reason. Okay, so we are done with the experiments. A part of experiments are the experimental designs, and I will discuss that once we're done with all these other research method techniques. So moving on to the second research method technique, we have observation. Now, observation is simply a study where we are observing the behavior of the participants, be it a human or an animal participant. So we're just observing them. For example, the study by Fagan, the behavior of the elephants are being observed. And this technique does not have an IV, it does not have a DV because as we discussed, an IV-DV is only a feature of an experiment. So if you do see an IV-DV during an observation, it's because you're doing an observation within an experiment, which is something that we can do. We have observations in Bandura, which is an experiment in Pilyavan, in what else? In Milgram. Any other study where we have Shackler, sorry, not Shackler, that's a part of the old syllabus, Dement and Kleitman. So we have lab experiments or field experiments where we can use observations, but the IVDV aspect is due to the experiment, not the observation. And I gave the example again of the elephant learning study by Fagan. It's not an experiment because there is no IVDV. So instead we consider it an observation. Now, what are the types of observations? We have a covert observation, which is when the participant is unaware that they're being observed. So in a covert observation, the observer is hidden. The participants do not know that they're being observed. And this could be done in numerous ways. Perhaps the observer is a stooge, a confederate, pretending to be a participant, and they're secretly observing you. So maybe in your classroom, one of the students is actually part of the research team dressed up as a student and is secretly observing you without your knowledge. Maybe a camera is secretly placed in the classroom and observers are observing you from the camera. So when the participants do not know that they're being observed, we call it a covert observation. However, when they do know that they're being observed, it is an overt observation, when participants are aware that they're being observed by an observer. Now, a participant observation is when the observer is part of the group that they're observing. So when we say that they're part of the group, it means that they are close to the participants. They are involved in the procedure. And again, an example of this could be the observer or the researcher who is pretending to be a student. So he's dressed up in the school uniform, pretending to be a student, and he's sitting in the classroom and he's observing everyone. So yes, that is a covert observation, but it is also a participant observation. And all these different observation techniques can overlap. So a participant observation is, again, when the participant or when the observer is part of the group that they're observing, and this can be either overt or covert. So in the case of the stooge observing the students in the classroom, it can be considered a covert participant observation. If, let's say, the teacher is in the classroom and is observing the students, and the students know that the teacher is observing them. So it's overt and also participant because the observer is close to the group. They're part of the setting, they're part of the group that they're observing. So that would be an overt participant observation. So you can overlap. A non-participant observation is when the observer is away from the group from that they're observing, away from the participants. This could be behind a one-way mirror. A one-way mirror is, if you've seen those interrogation rooms where there is a mirror and people outside the room can look inside, but people inside the room cannot look outside the mirror or the window. So it's an obstructed view. Only one side of the window or the mirror is allowing you to see through. So if there are observers behind the one-way mirror, they can see the participants, but the participants cannot see them. So because they're away from the group, they're at a distance, even through a video camera that is in the classroom or the setting, and you're not part of the group, that would be a non-participant observation. And similarly, this can also be overt or covert. If you do know that someone is observing you from a distance, it's an overt non-participant observation. If you are unaware that someone is observing you from a distance, then it is a non-participant covert observation. Then we have a structured observation. This is the Fagan study, as well as the Bandura study. Bandura is also an experiment, but it's also using a structured observation. A structured observation is one where the researcher is using something called a behavioral checklist, which is a list of behaviors that we want to observe. So for example, I want to observe how aggressive my participants are. So my study is on aggression and I want to observe their aggression. Now, aggression can be shown in many different ways, but I'm only interested in noting 5 particular aggressive behaviors. And let's say those are punching, kicking, shouting, scratching, and slapping. So these are 5 behaviors of aggression that I have noted down. I have a checklist and I've noted down these behaviors in that checklist, and I'm simply going to see how many times or whether or not the participant is showing any of these behaviors according to the checklist or not. So that is a structured observation. I'm using a checklist. I'm not interested in any other behavior that the participant might show. So any other aggressive behavior will be ignored. The focus is entirely on the behaviors that I've included in my checklist. An unstructured observation is one where there is no checklist. So if I'm observing aggression, any aggressive behavior will be recorded, and I'm not restricting it to 5 or 6 behaviors on a list. A controlled observation is an observation done in a controlled environment, so high levels of controls. This does not have to be a lab experiment. It can even be done in a field. Pilyavin is a field experiment, but it's also a controlled observation because they were observing the participants in a very controlled environment. Even the study by Fagan is considered a controlled observation. Milgram is considered a controlled observation because participants are being observed in a very controlled environment. And a naturalistic observation is when we're observing the participants in a natural environment with little to no controls. For example, if you are observing, let's say, animals in the jungle in the natural habitat, you're not controlling the environment in any way, it's completely natural, and you're simply observing them, that would be a naturalistic observation. Now, having discussed the definitions of each type of observation, let's go over the strengths and weaknesses of each. So, a covert observation, the strength of a covert observation is because participants do not know that they're being observed, there are fewer chances that there will be demand characteristics. Because naturally, if you do not know that someone is observing you, there is a greater chance that you will behave naturally, you will show your authentic behavior. So demand characteristics will be reduced, which will increase validity. So that is a strength. A weakness, however, is it can be unethical because there may not be consent. So informed consent, the guideline of informed consent would be broken because you are secretly observing someone without their permission. The strengths and weaknesses of an overt observation are exact opposite of covert. So there can be demand characteristics, which can lower validity because participants now know that they're being observed. But at the same time, if they know they're being observed, then they've probably given their consent. So it can be ethical in terms of consent. Now, a participant observation, the strength of a participant observation would be that the observer is close to the group that they're observing. So when you are close to the group you're observing, you're able to gather more detailed information. So a more detailed analysis or observation can be made of the behaviors. This is a pretty practical point because if you're close to someone, you're able to observe their behavior much better, more closely, and more accurately. So fewer chances of any mistakes being made. So again, a higher chance that your observation is accurate, is valid. But the problem with a participant observation is that particularly if it's overt, and the participants know that they're being observed and you're really close to them, then again, there are high chances of demand characteristics which can lower validity. Because naturally, if someone is very close to you and you know they're observing you, you might change your behavior. Okay. Non-participant would have the opposite participant. That's not how you spell it. I'll just write non-par. So non-participant, the opposite. When you're far away from the group, especially if it's covert, there are few chances of demand characteristics. Participants will show more natural behavior, hence increasing validity. But the weakness of a non-participant observation would be that if you're away from the group, you may miss out on some behaviors. You may make some mistakes because you're not close to them. So you might miss out on some behaviors. You might make some mistakes during your observation. Okay, then we have a structured and an unstructured observation. So let's talk about the strengths of a structured observation. So there are a few strengths of a structured observation. The fact that we're using a standardized checklist means that we can replicate it for reliability. Replicate it for reliability. Because it's a standardized checklist. A researcher has made this checklist. If we want to repeat the study, we can use that exact same checklist and assess for those exact same behaviors. So it can be repeated easily for reliability. Secondly, we are getting a lot of quantitative data, objective quantitative data, because we are observing the number of times that the participant is showing each of these behaviors. So how many times they displayed it, whether or not they displayed it. So it's giving us a lot of quantitative data. And the third strength of a structured observation is if we're looking at very specific behaviors, there is a good chance that we may not miss out on them. I'm interested in only 5 or 6 behaviors that are on my checklist. So I will be extra vigilant to make sure that I am observing these behaviors in case the participant is displaying any of these behaviors. Because if you're looking at any behavior whatsoever, if you're looking at multiple behaviors, you might miss out on some, you might note some. But if I'm restricting to myself to 5, 6 specific behaviors, I'm going to be more active in my observation. I'm going to be more focused in my observation, so I know that these particular behaviors are shown or not. Weakness, however, of a structured observation is that you are restricting yourself to some specific behaviors. What if the participant is showing other behaviors which could be very important, but because they're not in your checklist, they're being ignored? So that restriction, that limitation you're setting on yourself could perhaps lower validity. Unstructured observation is the opposite. The strength is that you're getting a lot more qualitative data because you're not setting restrictions on your observation. Any behavior that is relevant is going to be recorded. But the weakness of that is that it's not standardized, so it cannot be replicated. And like I said, if you're looking at multiple behaviors, many behaviors, you can make mistakes. You might note some, you might not note others. So there is more room for error in unstructured observations. And lastly, for a controlled and naturalistic observation, it's pretty much the same points as it would be for a lab and field. So in controlled observation, you can expect lower ecological validity, but higher levels of control, so greater chance of replicating it for reliability, higher standardization. And naturalistic observation, you can expect higher ecological validity and mundane realism, but fewer controls, so therefore more difficult to standardize and replicate for reliability. The next research method technique we'll do is self-report. And self-report basically is the way in which a participant is providing information themselves to the researcher. And this can be done through 2 ways, either through interviews or through questionnaires. Now, an interview, the definition is when a participant responds to questions in verbal form, whereas a questionnaire is when participants respond to questions in written form. And within interviews, we have 3 types. We have a structured interview, an unstructured interview, and a semi-structured interview. Now, a structured interview is when all participants are asked the exact same questions in the same order. So the researcher has a preset list of questions which they've already decided, and all participants are going to be asked those exact same questions, and they're going to be asked those same questions in the same order. So if I have 10 participants, and I have 10 questions that I've already decided, all 10 of those participants will be asked those same 10 questions in the same order. That's why it's called a structured interview. And I will not ask any additional questions. I'm going to stick to not the checklist, but the list of questions that I've made. A lot of students confuse structured interview with structured observation. There is a difference. In a structured observation, we have a behavioral checklist. And in a structured interview, we have a preset list of questions. So what students end up doing is they end up using the term behavioral checklist for structured interviews, and naturally they would lose marks. Just remind yourself that it's an interview. So we're not looking at behaviors, we're looking at responses. So you can't have a behavioral checklist, okay? An unstructured interview is when there is no list of pre-decided questions. So the participants are asked one basic question, and then the following questions are based on the answers to the previous question. So if I have participants that I'm interviewing, and I start off with one basic question, such as what is your favorite study in AS psychology? And one participant says it's the Milgram study, whereas another participant says it's the Perry study. So the person who said that Milgram is my favorite study, for them, my next question would be based on this answer. My next question could be, did you find Milgram to be too unethical? Whereas the person who chose Perry as their favorite study, for them, my next question could be, would you consider yourself as someone with high or low, or would you consider yourself someone with high levels of empathy or low levels of empathy? So you see, my questions will be based on the answers to previous questions. So there is no fixed set of questions that I've already pre-decided that I'll be following. So there is more room for me to ask open questions, to come up with new questions to assess the participants' responses. A semi-structured interview is a combination of both, where you've decided some questions that you will ask beforehand, but then other questions that you will ask in between, depending on the answers of the participants. And most job interviews are semi-structured, so all applicants are going to be asked a certain set of questions that the interviewers have already pre-decided. But within the interview, the interviewers can also ask additional questions based on the responses of the participants. So that would be semi-structured. Okay, again, strengths and weaknesses of each. Now, the general strength of an interview is that it gives you qualitative data, because participants are responding verbally. They're giving their opinions, they're giving their comments. So naturally, we're getting a lot of in-depth, detailed, qualitative data. Now, when we talk about specific types of interviews, a structured interview is standardized. Similar to structured observation, a structured interview is standardized. We are replicating the study, we're replicating the interview on all participants. I have a list of questions. I've asked one participant. I'm repeating those same questions on everyone else. So it's standardized, you can replicate it for reliability. The weakness, however, of a structured interview is that, so I'll write a tick for strength and an X for weakness. So a weakness of a structured a structured interview is that you cannot ask additional questions. So you are limiting or setting a limit to yourself in terms of the questions you can ask. So you're kind of reducing the amount of qualitative data that you can gather, because you want to ask maybe additional questions, but you cannot, because you're restricting yourself to just those specific questions that you've set in your list. For an unstructured interview, the strength is the exact opposite. We can ask additional questions, so we can ask a lot of open questions. It's giving us a lot of qualitative data, additional important qualitative data that could be very useful to us, but it is not standardized, because the questions that I'm asking will be different for everyone. So it's not easy to replicate for reliability. And of course, semi-structured will have the strengths and weaknesses of both. So it's a combination of both. So it will naturally have the strengths and weaknesses of both. Another general weakness of interviews, irrespective of whether it's structured, unstructured, or semi-structured, is that people can give socially desirable responses. So that's a problem with any self-report. People can give untruthful responses, they can lie, they can change their answers, they can answer according to what they believe is socially acceptable, which can lower validity. And another problem with interviews in general, irrespective of whichever technique you're using, is it can be time consuming because it's one-on-one. And people do not have the option of being anonymous. The interviewer is facing the person that they're interviewing. So it's face to face. So yes, there is the element of qualitative data, but again, it is time consuming and the participant does not have the option to be anonymous. And if they're not anonymous, then they may be a little hesitant to answer truthfully, which is why they may give socially desirable responses, which can lower the validity. The second type of self-report are questionnaires. As I said, these are when we respond to questions in written form. And there are two types of questions that we can ask: open questions or closed questions. Open would also be called unstructured, whereas closed would be called structured. Open questions are, and remember, this is no longer face-to-face now, this is in the form of a questionnaire. So it's written. Participants are writing their responses rather than verbally saying their responses. So an open question is when the participants can answer freely about a topic. We are not restricting their options, we're not restricting their responses. For example, what makes you happy? Okay, so that is an open question. You can write whatever you want. How do you feel about psychology as a subject in A levels? So you can answer openly without any restrictions. Closed questions, you are restricting the responses. So a closed question would be multiple choice, rating scales, or even scales in the form of statements, which we call a Likert or Likert scale. So you've probably taken some tests like personality tests, and you've seen that the options are agree, disagree, neutral. So you have to pick from these options. You cannot answer on your own. You have to select one of the options given to you. So when you have statements such as agree, disagree, neutral as the options, this is called a Likert scale. But let's say you are given options in terms of numbers. How happy are you? 1 to 10, 1 to 5. That is a rating scale. But either way, you do not have the option to answer openly. You have to answer according to the options given to you. So that is an example of a closed question. Now, the overall strengths of questionnaires are that they are standardized because everyone is taking the exact same questionnaire. So we can easily replicate it to test for reliability. Another point about the strengths of questionnaires is there may be the option of being anonymous. And if that is the case, then there are fewer chances of participants giving socially desirable responses. They may answer more truthfully if they have the option of being anonymous, which can increase validity. However, and also, yes, they're more convenient. You can get a lot of data in a very short time period, especially, is that how you spell convenient? Especially compared to interviews. Because interviews are one-on-one. Questionnaires you can hand out to hundreds of people and get information almost immediately. Now, the weakness of a questionnaire is, yes, I said if you have the option of being anonymous, it may reduce socially desirable responses. But again, with any self-report study, be it interviews, be it questions, there is always the problem that participants even then may answer untruthfully. They may still give socially desirable responses. They may still not give their actual answers, their actual opinions, which can lower validity. This is always going to be a potential problem with any self-report study. Now, specific to open and closed questions, if we are asking open questions, we are getting qualitative data, but we're not getting quantitative data. However, if we're only asking closed questions, then we're getting a lot of quantitative data, but no qualitative data. So it's always good to have a mix of both, so we can get both quantitative and qualitative data. Now, earlier on in reliability, I was talking about 2 points, the split half method and test-retest reliability. And I said that these are forms of reliability which will come in handy when we talk about questionnaires. So let's discuss exactly what those are. So I'll write them down over here. We have one technique called the split half method, and the other is called the test retest reliability method. Split half method is one way of assessing for consistency or reliability in questionnaires. So if I want to see if there is consistency in your answers on a questionnaire, I can split the questionnaire into two halves. This is not a very, I don't even know how to describe or what word to use for it, but it doesn't seem like a very, what's the word? It's a very simple technique, and for lack of a better word, it's a very silly technique of assessing for the reliability or consistency of answers in a questionnaire. And you'll know when I explain to you what it means or how it's being done. Let's say that there's a questionnaire with 20 questions. So what we can do is, I want to see if you have answered those 20 questions consistently. Okay, do we have consistent responses in those 20 questions? Because like some personality tests, if you've ever taken, you don't want the first half of your questionnaire to be answered with you responding as if you are showing an introverted personality style, but then the other half you're showing an extroverted personality style, because then there is obviously no consistency in your answers. So if you're an introvert, we're expecting answers accordingly throughout the questionnaire. If you're an extrovert, we're expecting answers accordingly throughout the entire questionnaire. So to assess for that consistency in your questionnaires, we can split the questionnaire into two halves. And you can do that by splitting into the first 10 questions and the second 10 questions. That's why it's called the split half method. We will go over your responses for the first 10. We will go over your responses for the second 10. And if there is similarity in your answers for the first half and the second half, then we can say that there is overall consistency across your entire questionnaire. So that is an example of the split half method. Another way is to split the questionnaire into odd and even numbers. So answer 1, answer 3, 5, 7, 9, etc. And then the even numbers, 2, 4, 6, 8, 10. And if your answers on the even set of questions and your answers on the odd set of questions have similarity, have consistency, then I can say that your overall questionnaire responses are reliable. They are consistent. You don't have to worry too much about this technique. It is something that you can consider if a question comes in paper 2 about one way in which we can assess for the reliability of responses in a questionnaire. Just simply say that we can use the split half method, where the questionnaire can be split into two halves, and the responses of the first half can be compared to those of the second half, and we can test for consistency. Okay, this is something which I will discuss when I make the video on paper 2, how to answer paper 2 questions. So I'll elaborate more on this. The other technique is the test retest method. Let's say that you take an IQ test, because that's a question, right? So you take an IQ test and your score comes to 120, which is above average intelligence. Now, if I give you the IQ test again, you should get the same score. If you get a higher or lower score, then I don't know which one was correct, the first time you took it or the second time you took it. So test retest reliability is when you take a questionnaire more than once and you get the same responses or the same results every time. So it shows that this is a valid representation of your behavior. You got 120 the first time, you get around 120, 121 the second time as well. So that means that you're showing consistency and your IQ is probably actually at that level. So that is test retest reliability. The next research method technique is case studies. Now, a case study is a study which is being conducted on one participant, and it is being done over a long time period. Let's say around 6 months at least or more. It could go on for 1 year, 2 years, maybe even 10 years. But it's being done on one participant. And an example of a case study is the study by Saavedra and Silverman on the boy who has a phobia of buttons. Again, there is no IVDV, but within a case study, it's common that we use other research method techniques to collect data, such as self-report or observations or both. Like the button phobia study, they interviewed the child and the mother. They used rating scale as well to collect data. But there was one participant and it was done over a long time period. So that is a case study. And another type of research method technique is a correlation. Now, a correlation is a study in which we are testing the relationship between two variables. And these variables are not IV and DV. Okay, so they do not have a cause and effect relationship. So what is the relationship that we're looking for between these two variables? Either a positive correlation or a negative or inverse correlation. A positive correlation is when the two variables are increasing or decreasing together. Whereas a negative or inverse correlation is when the two variables are moving in opposite directions. And let's look at an example of this to help you understand further. Let's say there are two variables that I want to see the correlation of: height and weight. And I'm testing for a correlation. I'm not looking for, I'm not conducting an experiment, because if I do an experiment, then I'm saying height is the IV and weight is the DV. And it's because of height that weight is increasing or decreasing. So that's cause and effect. That is not something that I'm assessing in a correlation. So you have to be very careful about this difference between correlations and experiments. Okay, there is no cause and effect. We are not saying that height is the reason weight is increasing or weight is the reason that height is increasing or decreasing. They are two separate independent variables, but they're not affecting each other. We're just looking for a general relationship between the two, if they both increase or decrease together or move in opposite directions. So let's say I have a sample of 5 people and I measure their height and I measure their weight. So let's say someone is around 6 feet tall and their weight is 80 kg, 80 kilograms. Then another participant is 5'11 and their weight is 75. A third participant is 5'9 and their weight is 70. The fourth one is 5'5 and their weight is 60. And the last one is 5 feet tall and their weight is 45. Now, what is the trend that we're seeing here? As the height is going down, we can see the weight is also going down. Similarly, you can look at it the other way around. As the height is increasing, the weight is increasing. So both height and weight are moving in the same direction. This is an example of a positive correlation, where both variables are moving in the same direction. But again, I'm not saying it's because of height that the person's weight is more or less. The moment I say that, this is no longer correlation, this is an experiment, because then I've automatically turned height into the IV and weight into the DV. So we have to be careful of not doing that in a correlation. Okay. So within a correlation, just like in a case study, if you wish to collect data, you can do so by interviews or by observation. Like over here, I'm observing their weight, I'm observing their height. I can even interview them. I can even give them questionnaires in which I can ask them questions about their height, their weight, so that I can use those as measures to collect data and compare the results to identify whether there is a positive or a negative correlation. Now let's take an example of what could be a negative correlation. So let's say I want to see a correlation between two variables. Let me think of something. How about time spent using social media? So time spent using social media. So this is one variable. So variable one. And happiness. So how happy you are in life in general. Now let's say that the results of 50 participants are that overall those who spend more time on social media are less happy. So I'll give them a questionnaire, let's say, and I'll ask them how much time on average in a day do you spend on social media. And then I can give them another questionnaire in which I can ask them to rate how happy are you on most days from 1 to 10. And then I can compare the ratings on both the measures, and I can see what the link is. And let's say that those who spend more time also rated themselves as being less happy on the questionnaire, then I can see that there is an inverse correlation, because when variable 1 is increasing, then variable 2 is decreasing. So inverse correlation, negative correlation. But again, I am not saying that it's because of the usage of social media that you're happy or not happy, because then that would be an IV and DV. I'm just saying that there is a relationship, but not of cause and effect. Okay, so this is a correlation. Let's say there are two variables, intelligence and height, and there is no correlation. So that is also possible. Maybe there is no correlation. Maybe someone who's tall is very intelligent. Maybe someone who's short is very intelligent. Someone whose average height is also very intelligent. So that could be anything. So when there is no relationship between two variables, we will say there is no correlation. Now, there is a way of assessing how strong the correlation is between two variables. And for that, we use something called a correlation coefficient. We'll just write that down. A correlation coefficient. Now, you do not need to know how this is calculated, but you should know what it is. It is basically a number from -1 to +1. The minus or plus sign is indicating whether it is a positive correlation or a negative correlation. And the number is indicating whether it is a strong correlation or a weak correlation. The closer the number is to 1, the stronger the correlation. Now, remember, you do not need to know how it's calculated. You should just know what it means. Let's say that the correlation coefficient of height and weight is calculated as 0.97. Okay. Now, there's no sign, then we're assuming that's a plus, positive. So, because if this was minus 0.97, then we know it's a negative correlation, but it's 0.97. So because it's a positive number, it's a positive correlation. So immediately we know that height and weight will either increase or decrease together. But how strong is that relationship? That is depicted by this number, 0.97. 0.97 is very close to 1. Now remember, I told you the closer the number to 1, the stronger the correlation. The closer the number to 0, the weaker the correlation. 0.97 is very close to 1. So this is a very strong correlation. You can almost consider this as saying there is a 97% chance that if you're tall, if the height increases, then your weight also increases. Or if height decreases, then weight decreases. Okay. Let's say the correlation coefficient of time spent using social media and happiness comes to be minus 0.65. So this is a negative correlation because of the minus sign. So we know that if you spend more time on social media, then happiness will decrease. So, and vice versa. But how strong is this correlation? Only 65%. So it's above average, but it's not very strong. It's slightly above average in terms of its strength. So there's a 65% chance that the more time you spend on social media, the less happy you are. Okay. If the correlation coefficient of two variables, let's take another example. I'll just write two random variables, variable 1 and variable 2. So let's say variable 1 and variable 2 have a correlation coefficient of 0.5. So it's a positive correlation, but an average strength correlation. Let's say it's minus 0.5. So an average strength correlation, but negative. If two variables have a correlation coefficient of minus 0.10, it's a negative correlation, but a very weak correlation, almost non-existent, because 0.10 is very close to 0. So a very low chance that if one variable increases, the other will decrease. Okay, so I hope you understand the definitions of correlations and how to understand what the correlation coefficient is. And an example of this is the Baron-Cohen study. the eyes test and the AQ test, they had a negative correlation of minus 0.53, which means that there is a 53% chance that if one scores high in the eyes test, they will get a low score in the AQ test. So there is an inverse correlation, but not a very strong one. And that is true because in the Baron-Cohen study, the results did show that there was an inverse correlation overall. But when I took the two tests, I actually scored high in both. So there is no guarantee that you will always score high in one and low in the other. There are cases where some people might score high in both or low in both, which is why it's not a very strong correlation. It's 0.53%, 0.53 correlation, 53% correlation, but an inverse one. Okay, let's talk about the strengths and weaknesses of the two research methods that we've just discussed, case study and correlation. So a case study has one participant, and we're doing a study over a long time period. So let's talk about case study. So one strength of a case study is that we get a lot of in-depth, detailed, qualitative data. Because we're studying a participant for such a long time, and we're using techniques such as interviews to collect data, self-report to collect data, observation to collect data, we're getting a lot of data. So a lot of qualitative data is being collected. A second strength of case studies are we can measure or observe the development of the participant's behavior over time. Because if you're studying someone for, let's say, 5 years, 10 years, you can see how their behavior is changing over time. If someone is coming to me for a phobia, for example, the button phobia study, we can see how much his phobia was at the beginning of the study, then they saw it 6 months later, and then they saw it 12 months later. So over time, you can see how his phobia is decreasing or increasing. So the changes or the development in the participant's behavior can be measured over time. And thirdly, actually, no, we'll get to these weaknesses now. So one weakness of a case study is a very obvious weakness is that there is little to no generalizability. Because you're doing a study on one single participant, so you cannot apply the findings to anyone else. No one else is being represented. There is a possibility that the participant and the researcher may develop an attachment or a relationship. And for that reason, the researcher could then make mistakes in interpreting or analyzing the participant's behavior. So there can be a possibility of researcher bias if you, over time, get to know the participant and if a certain bond or relationship develops, which can lower validity, obviously. Number three, there is a chance of participant or sample attrition. Let me just write that again. Participant, ATTRITION, sample attrition. This is when the participant exercises their right to withdraw and they do not wish to continue. Your plan was to do the study for 5 years, but after 2 to 3 years, the participant decides to leave the study. And you cannot force them to continue. So because you only have one participant and they've withdrawn, or let's say they fall sick or they leave town, or maybe they even pass away, your study will be left incomplete. So these are the strengths and weaknesses of a case study. Now let's talk about the strengths and weaknesses of a correlation. So a correlation, as you saw, we are collecting data which is mostly quantitative. So there is a lot of quantitative data that is being collected. They are more ethical to conduct compared to experiments. And we are able to determine the relationship between two variables. But the weakness of a correlation is that we cannot determine cause and effect. So it's a definition of it, but it's also a weakness that cause and effect between two variables cannot be determined. And the last research method technique is a longitudinal study. Now, case studies are longitudinal, but other research method techniques can also be longitudinal. You can have a longitudinal experiment. You can have a longitudinal self-report study. It basically means doing any of the other 5 research method techniques, but over a long time period, for a year, 5 years, 10 years, etc. And it's being done on the same participant over time. So the participant isn't changing during this time period. So you can do the study in intervals if you wish. You can do it um, every 1 year if you wish. You can do it. So you can decide the way in which you want to do it. But the idea is that it's always the same participant or the same set of participants, and they're being studied over time. Um, the participants' behavior can change over time, their age can change over time, and obviously what this does is it's allowing the researcher to study these changes in time frames such as over months, over weeks, over years, etc. Now, cross-sectional studies are different from longitudinal studies. Cross-sectional studies are basically studies that are done on different groups of participants at one point in time. So in longitudinal study, it's the same set of participants, but in a cross-sectional study, participants are changing. So maybe you did a study on one group in 1 month, and then the next month you did it on a separate group. So you're not looking at the changes in the same participants' behavior over time. Instead, you're comparing different groups of participants to one another. Okay, a strength of a longitudinal study is that we are sure that any changes in the participants' behavior is purely because of changes over time. Because the participant isn't changing. So any changes in development we know is due to passage of time. And the fact that it's the same participant over time, participant variables are being reduced. It's always the same participant. So this can increase validity. Weaknesses, however, is that generalizability is low because it's the same group that is studied over a long time period. And similar to case studies, if you're studying a set of participants over time, an attachment or bond can develop, which can cause researcher bias, which will lower validity. And again, similar to case studies, if participants decide to withdraw sample attrition, then that is a problem because you are not changing the participants, it's the same set of participants. So if some of them decide to withdraw, then that could also affect your study. Other points about weaknesses could be that doing a study over a long time period can be difficult to standardize, so reliability could be an issue as well. Okay, now experimental design is a feature of experiments. That's why it's called an experimental design. It is the way in which participants are being exposed to different conditions or different levels of the IV. And as we discussed earlier, we normally have an experimental condition and a control condition. So the way in which participants are exposed to these two different conditions is called the experimental design. And there are three types we should know: independent measures design, repeated measures design, and matched pairs design. An independent measures design is when there are different participants in each level of the IV. Repeated is when the same participants are repeated in all levels of the IV. And the matched pairs design isn't a main experimental design, but it's like a secondary experimental design that we can use with independent measures in which we match the different groups in terms of certain variables. Now let's try to understand this with examples. So let's take the example of the experiment we were doing at the beginning of this lecture. We have 30 participants and 30 participants are split into two groups. We have the AC on group, 15 participants here, and we have an AC off group, 15 participants here. AC is the IV and concentration is the DV. So the two conditions that we have are AC on, which is the experimental condition, and the control condition of the AC off, and 15 participants in each. Now, an independent measures design is when there are different participants in each condition of the IV. So what are the two conditions of the IV? AC on, AC off. Do we have different participants in each group? Yes, we do. These 15 are different, these 15 are different. So we have different participants in each condition of the IV. These 15 are exposed to the AC on, whereas these 15 are exposed to the AC off condition. This is an example of an independent measures design. And we can even call this an independent groups design because there are two independent groups, two separate groups with different participants. These guys are only taking part in the procedure once, where the AC will be on. These guys are taking part in the same procedure once, but with the AC being off. And what is the procedure? That they're in a classroom and they're taking a test, and their concentration levels are being assessed, or their performance in the test is being assessed. Okay? So this is an example of an independent measure design. A repeated measures design is when the participants are being repeated in both conditions of the IV. So in order for this to be a repeated measures design, all 30 participants would be tested in the AC on condition where they would take the test, and then maybe the next day or a few hours later, or up to the researcher, the participants, all 30 of them, will be tested in the AC off condition. So they will take the test again, but this time in the AC off condition. So they're being repeated in both conditions of the IV. Students in repeated measures design, for the definition, they end up writing that the participants are repeated in the procedure. That is not the correct definition. Participants can be repeated in the procedure in an independent measures design as well. What if I have 15 participants in the AC on and a separate 15 in the AC off, and I do the study 3 times? So technically they are being repeated. They're doing the same study 3 times, but those 15 that are doing it in the AC on are always doing it in just that level of the IV. And those 15 doing it in the AC off are always doing it in the AC off level of the IV. So it doesn't matter how many times you do the procedure, it will only be an independent measures design if you are always doing the study in which you're exposed to one level of the IV. But the moment you are tested in both conditions of the IV, the experimental and the control condition, then it is automatically a repeated measures design. So in what example can I give? In the Andrade study, there are 20 participants who are in the doodling group, and there are 20 participants in the control group. This is an independent measure design. If this was to be a repeated measure design, all 40 would do the be part of the doodling condition, and then they would do the entire procedure again, but this time with the control sheet of paper. So they're being repeated. So that would be a repeated measure design. And each of these have their strengths and weaknesses. A strength of an independent measures design is that because there are separate groups, so let's take the example of an independent design. So let's say again 15 here and 15 here. So this is an IMD, independent measures design. Now the strength of an independent measures design is that there are no individual differences. Oh, sorry, there are no order effects. Because you're taking the test once in the AC on condition, the other group is taking the test once in the AC off condition, so order effects are being eliminated. So remember the slide about order effects, ways to overcome them, randomization, counterbalancing, and then the third point was using an IMD. This is why independent measures design can overcome order effects, because you're only taking part in the task once. A weakness, however, so this is a strength. A weakness, however, is that there can be individual differences. There can be participant variables, which can lower validity. Now, how can they lower validity? We discussed that what if in one group, students are academically smarter? What if in one group, students just generally have better concentration levels? What if in one group there are students who have ADHD? Like 5 students have ADHD, so obviously their concentration levels are already very low. So these are individual differences between two groups, in which one group may have an advantage over the other. So technically confounding variables, participant variables, which can lower validity. Okay. Now, one way to overcome this is to do a matched pairs design. So a matched pairs design is a way of keeping an independent measures design while removing individual differences. So in our study, we identified that maybe if you're allocating them randomly, then one set of participants might have students who are academically superior than the other. So what did we do? We checked their grades and we split them evenly. We made sure that there are an even number of students with an A, a B, a C, a D in the AC on condition, and an even number of students with an A, B, C, D in the AC off condition. So I have matched my two groups in terms of their academic level. So that particular individual difference is no longer a confounding variable in my study. And it is still an independent measures design, but removing this problem that was lowering my validity. And you can match your groups in terms of many variables. You can do it in terms of age, you can do it in terms of gender, in terms of IQ. It depends on whatever you feel is necessary. So a match-based design helps you to remove individual differences, but it can be a little difficult and tedious to administer because you almost have to do an entire pre-procedure. You have to see their grades in this particular example, see who's gotten what grades, then allocate them evenly in the two groups. So that can be a little time consuming. As for a repeated measures design, it's the exact opposite strengths and weaknesses as those of an independent measure design. Weakness is that there can be order effects because if you're taking the test in the AC on condition and then you're taking the same test in the AC off condition, then there is a chance that the second time you already know the answers, so your performance will naturally be better. And it's got nothing to do with the AC being on or off, but due to practice effects. So that's a weakness of a repeated measure design, that there can be order effects which can lower validity. But the strength is that there are no individual differences. Because now you're not comparing your performance in the AC on to someone else's performance in the AC off. You're comparing your own performance in the two conditions. You're seeing if you did better in AC on or if you did better in AC off. So you're seeing which condition affected your concentration more. So individual differences are being removed because you're not being compared to someone else. You're being compared to your own self in both conditions of the IV. And the last topic are the sampling methods. So this is the way in which we can get participants to take part in our study, how we acquire participants, because naturally in order to do a study, we have to have participants. So sample, participants, all mean the same thing. A target population is basically the population of people on whom you wish to do the study. So if I want to do a study on students of a particular school, and let's say that school has 1,000 students, so those 1,000 students would be my target population. And my sample would be, let's say, 50 students who actually represent that target population who are taking part in my study. So the people who are actually participating, who are part of the target population, is the sample. So they are my participants. Three ways in which we can acquire participants for a study: volunteer sampling, opportunity sampling, random sampling. Volunteer sampling is also called self-selecting. This is when participants volunteer or select themselves to be part of a study. And this is often done where researchers can place ads on social media or in newspapers. They can inform participants or potential participants that they want to do a study and that they would be paid for their participation, and then participants would volunteer to be part of the study. Now, a strength of this is obviously that they're giving consent, so it's ethical. But a weakness of this is that there can be demand characteristics. Okay. Another strength of a volunteer sampling is that you can get specifically who you require for your study. I only wish to do a study on females of the age 30 to 50. So I can specify that criteria in my ad, and only those people who meet that criteria will volunteer. So I can control the features of my participants. And another weakness of volunteer sample is, one was that it's possible that they may show demand characteristics. Another is that it can be time consuming and expensive. You have to wait for people to respond to your ad. It might be expensive to pay for the ad. You may even have to pay participants to volunteer, to give them some incentive to volunteer. So it could be time consuming and expensive. Opportunity sampling is also known as convenience sampling, and this is when participants are readily available at the time of the study. You go to a park, there are 50 people present there, you do the study on them. That's an opportunity sample. You're taking opportunity or advantage of the fact that people are already present at an area where you're wishing to do the study. Now, the strength of that is that it's very convenient. because you don't have to wait for people to show up. They're already present at the area where the study is being conducted, like the Pilyavin study. Whoever was on the subway, the study was done on them. In most cases, participants may not even know they're part of a study, so fewer chances of demand characteristics. Again, an example is Pilyavin. However, the weakness of that is that it can be unethical, because obviously then they would not be giving consent. And another weakness is, unlike in volunteer, you can't control the specific features of your participants. What if I go to the park and I do a study on 50 people, but all of them happen to be people above the age of 40, and most of them are males? So my generalizability is going to be limited to only that particular sample, not to younger people and not to females. And the third one is random sampling. Random sampling is when everyone has an equal chance of being selected at random from a target population. So let's say I said that I wanted to do a study on students of a particular school. So those 1,000 students are my target population. But I want to select only 50 people to actually take part in my study. So those 50 people who I will select, I can select them randomly. I can have names of people on chits of paper that can be folded and put into a bowl, or that can be put into a hat. And I can randomly take out any 50 chits, and whoever is selected is being selected at random. So that is an example of random sampling. None of the 12 studies are using random sampling. They're either using volunteer or opportunity. Some people believe that the Milgram study is random sampling. It is not random sampling. The role allocation that was done is not a sampling technique. That is something that was done in the procedure. The participants were acquired through volunteer sample. Okay, so make note of that. It's not a random sampling technique. The role allocation done was apparently said to be random, even though it wasn't, it was rigged. But that's not sampling. Sampling is the way participants are acquired for your study. It's got nothing to do with the procedure. And the strength of a random sampling is, irrespective of whoever you are choosing, you know that they're representing your target population. But the weakness is that it's not representing people outside the target population. So whoever the 50 people are, they are from the 1,000 target population of that school. So it is generalizable to them, but not generalizable to people outside that school. I said that there were two other sampling methods which are not very important, but if you wish to use them in paper 2, you can. One is stratified sampling, also known as quota sampling, and the other is snowball sampling. Stratified sampling is that you set quotas or stratas and you select a certain number of people from each strata. For example, I want to select students from 4 different schools: School A, School B, School C, School D. I've set a limit that I want 20 students from School A, 15 from B, 10 from C, and 5 from School D. So I've set quotas for each of these different stratas, each of these different schools from where I wish to select my participants. So this is stratified sampling. Snowball sampling is a form of word of mouth where I have, let's say, 5 participants and I ask them to recruit more participants. So they go and they tell 5 more. So now that makes it 10. Those 5 go on and recruit another 10. So that makes it 20. So this just basically goes on and on and on. So just like a snowball, you're adding more and more snow, it builds bigger and bigger. Participants are increasing in number by informing more and more people. So that is snowball sampling. Okay, a few other terms which you maybe should know. There is a concept called a double blind technique and a single blind technique. It's related to validity. Let's say I am testing the impact of a drug to treat depression. So the drug would be the IV and the depression would be the DV, because depression is dependent on, or the increase or the any decrease or improvement or reduction in depression is dependent on the drug that I'm giving you. So because we know that IV has to have at least 2 conditions. So what are the 2 conditions? The first can be an antidepressant, and the other could be a placebo. A placebo is basically like a fake drug, and mostly in the form of like a saline solution or water. So like a control, basically. So the antidepressant condition would be the experimental condition. The placebo would be the control condition. Now, a single blind technique is when participants are not aware which condition they're in. So I have 40 participants, and all of them are diagnosed with depression. 20 of them will be given the antidepressants, and 20 of them will be given the placebo, but they all think that they're getting the same thing. This is a single blind technique. Because if you know you're getting a placebo, if you know you're getting a fake drug, then obviously it will lead to demand characteristics, it will affect your behavior. So to try and avoid that, I will not let you know that you are in the placebo group. So this is a single blind technique when the participants are unaware what condition they're in. A double blind technique is when both participants and observers are not aware which condition the participant is in. So participants don't know if they're in the antidepressant or control condition, placebo condition, and neither do the observers. Now, why is it important for the observers not to know? If the observer knows that you are in the placebo condition, it may interfere with their observation. It may cause a bias. They may already be expecting you to not have any reduction in your symptoms of depression because they know you're in the placebo group, they know you're not being given the drug. So just to make sure that their observation is valid, is accurate, is authentic, at times even the observer is not told which condition the participant is in. So obviously they will know later on once the results are being analyzed, but during the observation process, if both participants don't know which condition they're in and the observers don't know, it increases validity. It reduces demand characteristics from the participants' end, and it reduces researcher bias from the observer's end. So overall, it is increasing validity. Okay, one last thing I want to discuss. So we have to go back to observations for this, specifically structured observations. So remember, a structured observation is an observation where we're using something called a behavioral checklist, right? A list of behaviors. Now, there's something called time sampling or observation sampling. Observation sampling is a way of us to record observations, record behaviors during an observation, particularly when we're using a checklist. So I have those 5 behaviors of aggression that I want to observe from the participants, but how exactly will I record them? That can be done through observation sampling. And there's one way of observation sampling, actually two. One is called time sampling, and the other is called event sampling. Now, within time sampling, we have 3 types. There is the predominant activity scan. There is the 1-0 scan, and there is the instantaneous scan. Instantaneous scan. In time sampling, you set a certain time period within which you wish to observe the participant's behavior. So I want to see if they show any of those aggressive behaviors that I've noted on my checklist, the screaming, punching, slapping, shouting, etc. Those 5 behaviors that I want to observe, I want to observe the participants for 20 minutes and I want to see if they show any of these behaviors. In an instantaneous scan, what I'm doing is I'm setting intervals. This is something Bandura also did. So for 20 minutes, I can note down or I can record my observations after every, let's say, 10-second intervals. So 10 seconds, I note the behavior. If they showed any of the behaviors on the checklist, I'll make a note of it. Then I'll do the same thing after another 10 seconds, then another 10 seconds, then another 10 seconds, all the way up to the 20 minutes. So this is called an instantaneous scan. This is something that Bandura did. If you don't wish to use the term instantaneous scan, you can just say time sampling was used. Another form is called 1-0 scan. 1-0 scan, you set a time limit, let's say again 20 minutes, but you simply state if the behavior was shown or not. So you're not saying how many times it was shown, you're just simply stating if the behaviors on your checklist were displayed or not even once. And predominant activity scan is you set a time limit, let's say 20 minutes. You've got 5 behaviors you want to observe. You will note which of the 5 behaviors was the predominant activity, which one was the one that was displayed the most by the participant within those 20 minutes from those 5 behaviors. Then we have event sampling. Event sampling is also setting a time limit, but we record the number of times that a certain behavior was displayed. So the event of punching, the event of screaming, the event of abusing, let's say, we just tally the number of times each of these behaviors were shown within a certain time period of 20 minutes, for example, that you have set. So these are all different ways in which we can record observations during an observational study, especially when we're using a structured observation. So this is a rather lengthy video, but it's a very important video because it's research methods, and I tried to cover it in as much detail as I could. Again, I said that you can join the WhatsApp group. I'll send the link below in the description. Contact me, email me, WhatsApp me for any questions you might have. If you wish to get the PDF of the detailed notes on my research method slides, you can access those. So just message me, I'll send them to you. And this is part 1 of research methods. I haven't done data analysis as yet. I'll make another video on that. And then I'll make another video on operationalization as well as on hypothesis and aim, one-tailed, two-tailed. And once we've covered all of that, then I'll make a video on paper 2, the type of questions, how to answer them, what to expect, and how to design your own study. So thank you and see you soon, hopefully. Bye-bye.